Hello, everybody. We are so excited to have you today at the Science Lab Tech Open House. My name is Amanda Rosenzweig. I am the Assistant Dean of STEM, and the Science Lab Tech program falls under STEM. So I am one of the people that helps direct and run the program. We also have Miss Charlene Shunick on the call, who will be speaking in just a moment, and she is the biotechnology director. If you are not a student in our program yet, you can grab your phone and you can grab this QR code, and this will bring you to our webpage for SLT. And just so everyone's aware, this slide deck, along with the presenter's slide deck and the recording, will be sent out to all attendees. So if you can only stay for one session or you get to stay for them all, you will be able to watch back some of the sessions you may or may not be able to see. Also, you can take a moment if you want to follow us on social media. Miss Shunick and I are pretty active on Facebook, LinkedIn, and Instagram, posting all the cool stuff our students take part in, as well as opportunities for careers, internships. This information is also there. So again, this slide deck will be shared out with everyone attending. So I just want to talk a little bit about the program, the SLT program. So SLT Science Lab Tech is going to have two concentrations in it. It's going to have chemical technology and it's going to have a biotechnology concentration. To enter this program, there are no entry applications, but you do have to have four main prerequisites completed. And those are going to be your general biology lecture and lab for majors and your general chemistry lecture and lab for majors. Once these four classes are completed, you can then enter our Science 130 and 132 class. That is the entry class for the SLT program, and that is offered every fall and spring. After that, there's a lot of curriculum that you can take depending on which concentration you will fall into, and those can be found at the website, or you can look at these two links when the slide deck's provided. We've been very fortunate to have phenomenal students, and our students are really thriving, and we have 100% placement of our students into the workforce. Many of them are in the workforce and going to school also. So we have quite a bit of students all over the place. Some of them are outside of Louisiana. They end up moving to Minnesota, New Mexico, California. And they're going because a lot of it is through networking, through opportunities like this. As you can see, there's a lot of different industries that are hiring our students within the New Orleans greater area: Chevron, Dow, Shell, Abbott, Abatala, Tulane School of Medicine, Intertech Eurofins. Even one of our past graduates is our biotechnology lab coordinator, and she does all of the lab prep for all of the faculty members. We've had people at coroner's office, Axosim, LaSalle, all of those individuals, a lot of them do cell research. So a lot of cool things. Our students are also going, as I said, to get a bachelor's, master's, and PhD. So we have students that go to UNO, LSU, Tulane Medical School, and Xavier. So there's a lot of opportunities for our students after they graduate with their associates. What's even cooler is we are in curriculum right now to get certificate programs. So as you go through the curriculum for your associates, after 2 to 3 classes, depending on the certificate, you will also have certificates. These are stackable and they give you credentials. So as you are going through the program, you are more hireable and employable. Now, what is kind of cool is that many of our faculty are very up to date with industry trends because they also still work in industry. And we've been very fortunate to have a lot of consortiums and grants that support our research and our students' exposure to what goes on in real world situations. So one thing is that we work a lot with algae. We work with the Algae Technology Education Consortium and a company called SBI for algae research. We have a grant with National Science Foundation right now to integrate biomanufacturing practices, which is becoming huge in the greater Gulf South region. So it gives students exposure to more skill sets to help them get hired. We have a RISE grant through the USDA doing work on monoclonal antibody research with Dr. Nesbitt. We are biosafety level 2 laboratory, which means we can work with a lot more cell types. That is being approved this week or next week. So we'll be live with that. And then we also work with leading industry consortiums to expose our students to opportunities. So we recently had last month a biofab hackathon where students worked with industry leaders from Europe and the United States to create AI simulated work-based place scenarios. So we are really excited of the opportunities we get to expose our students to. We have relationships between many different partnerships throughout the greater Louisiana area, as well as nationally. These are some of our partnerships within town. And then these are some of our national engagement partnerships. So awesome opportunities for our students. Now, I'm not going to go through the rest of these slides, but I just want you to know the next few slides are introducing our faculty. So you can go read about our faculty. And then it talks about different opportunities our students have had, different opportunities we provide our students. And then the rest of it is going to be student testimonials. We've had 45 graduates, but there are just about 10 testimonials in here. So this is all information that will be sent to you. So we're really excited that you did join. But I'm going to give Ms. Shunick, sorry, I'm scrolling. I don't want to give anyone a headache. But I'm going to give Ms. Shunick an opportunity to speak about biotechnology, specifically as our chemical technology director. She had a family emergency, so is unable to attend today.
Hey, everybody. First of all, it's so great to see so many of my students here. Thank y'all for coming. So I know that I teach many of y'all in a lot of different things, although some of my forensic science students are here. And outside of my role as an educator at Delgado, I am the biotechnology director, and I've been in this position for, I think, 7 or almost 8 years now. I'm really proud of this program because we have such a variety of different types of skill sets, equipment, and classes that we offer. And our faculty are so well versed in these fields that they're able to pick up and update with the up and coming kind of techniques as they change. And we're able to ask them constantly about whether or not we should invest into a new piece of equipment and if they're going to be able to use it. So for y'all that sort of have heard of me, I am the person here that also offers all of the forensic science courses through the School of STEM. And I also have an animal forensic research facility, which is a fancy word for a body farm. And, biotechnology is a little bit of a tricky field to really explain because it is a marriage of science and technology. And our courses are really based on learning how to utilize different parts of biology and the equipment that's available to manipulate it. So some of the scarier things you hear about casually, like GMOs or like cell cloning and things like that, are realistic things that our students learn how to do and that our professors are able to do. And the coolest part is that the technology updates so quickly that teaching old school methodology only prepares students how to do the newer school things. And I'm really proud of our graduates because when they leave, they have skill sets that transfer to any laboratory space, not only in the United States, but across the world. So they're able to go and do a variety of different careers. And I know a lot of my students here are interested in going into medical fields, but this is kind of a cousin of medical laboratory technology. And some of our students have even gone to work in hospital environments and laboratory settings there testing things. So, the types of things that our students are studying is varied. The types of courses we offer are varied. And each of our professors kind of have a niche that they incorporate to the classes as they teach them, which is like the coolest part. Other classes with us, they're a little bit more rigorous. We have our textbook and things we have to kind of get through. Whereas these classes are a little bit more flexible in when we do things and how we do them and what we're going to use each semester. So it's really fun. A lot of students find that they actually really enjoy sitting at a bench and doing some of the type of work that is required to be done. And I personally think the coolest part of laboratory science, whether we're talking about chemtech or biotech, is that something you might work on the first month of a class is something you're going to use throughout the semester and end up finishing at the last portion of the class. So you kind of see how little errors and little mistakes that you make along the way come back to bite you later on. It's been really fun. I've learned a lot about biotechnology. And my colleagues have actually taught me how to use some of this equipment, which is in my area. I've been able to beef up my forensic science courses a lot. So now, we know how to do DNA extraction and purification and profiling and all kinds of other things that previously I didn't know a whole lot about. Um, our biotech students in particular have gone to quite a lot of different job fields, but also many of them have chosen to go to the bachelor, master, PhD path because they're interested in research. And for people who are thinking about that, the skills you get with us transfer over to those programs as well. And in fact, you usually have on par, if not slightly above the skill level, because our classes emphasize that. Unlike maybe the traditional lecture lab combo where you're learning about it in lecture and doing it in lab for 3 hours once a week, our labs meet for 3 hours twice a week. So you'll really learn how to get your hands dirty and troubleshoot and do all kinds of different methodologies in our classes. And for y'all that aren't sure, I always recommend taking the intro classes as electives, at least the lab, because you get your hands dirty and you get to sort of see if you're into handling, microscopic biological specimens and doing things to them. I personally think our tissue culture courses are like the bee's knees. They're like top of the field. And that does seem to be one of those skills that almost all laboratories in the area and in our partnerships require. So we really prepare y'all to be successful in your future careers. It's been so fun. And we just keep getting more and more fancy equipment as we need it. So we're constantly staying up to date. It's really cool.
Thank you very much, Ms. Shunik. Just to add to what Ms. Shunik said before we introduce our first speaker, we are constantly trying to stay up to date with industry standards and ensuring that our students are leaving our program with the proper skill sets, knowledge, soft skills. And one of the things we do that by is grants. And we are currently waiting to hear from the NSF for 2 more grants that we will learn about in May if we have received them. And both of those grants are directly related to the SLT program. So there is a lot of funding and financial support for us to continue growing, which in turn helps support student success and opportunities. So just be aware that this is a growing field. All of the predictive research from the government shows that this field is going to have about a 60% increase in needed employment, and we don't have the workforce to fill those. So not only is there a demand for you, but the pay is actually really great. In Louisiana, I hate saying it, it's not as great as around the nation, but we are in Louisiana. But if you are willing to move, there will be a lot of opportunities to earn a great salary with your associates if you choose not to go down that bachelor's, master's, PhD path. And I apologize, I forgot to turn my phone off.
I was going to add one more thing. at the end of this program, whether you're in the chemtech or biotech path, and sometimes students choose to take courses in both concentrations, just depending on, like, forensic science students sometimes need to take some chemtech courses. You do an internship, which I'm kind of like in charge of. And the internship has been a place where students gain confidence in the skills that they have, but often a place where they find either short-term or long-term employment as well, or opportunity to get into a program. So by the end of our program, you have had some job experience. We help you with your resume and cover letter to make you employable and kind of prepare to go into the workforce. So it's a really great opportunity for students who are coming here with the intention of changing their career and wanting to go right into the workforce. Our students have been extremely successful in doing so because of our internship program as well.
Thank you, Ms. Schoenig. So, with no further delay, I am going to introduce our first speaker who we're really excited to have. Our first speaker is Dr. Bruni, who has been a phenomenal supporter of our program. I do want to, before I introduce her, say she has hired one of our students and they wrote a paper. So the student is published now and is just thriving. The student's even on the website. I saw that previously last week. So awesome job to that student. But Dr. Bruni is a research microbiologist with the U.S. Department of Agriculture Research Service, which is basically USDA, which is right down the street from Delgado's main campus, City Park. And her work focuses on microbial research within the Commodity Utilization Research Unit, which she will explain all this. And this contributes to the National Agriculture Innovation. She holds her academic training from Tulane University and the University of Montana, and we are very grateful for her to continue supporting us and to be here to share her experiences with you. So I'm going to stop sharing and allow Dr. Bruni to continue.
All right, it's great to be with you today. Thanks for having me. Let me go ahead and see if I can get my slides shared here. I have just a few dozen windows open today. Let's see. Let me know when
we can see them.
Great. All righty. Well, like y'all said, my name's Gillian Bruni. I'm a research microbiologist over here with the Agricultural Research Service here at the Southern Regional Research Center in New Orleans. And I'm housed within the Commodity Utilization Research Unit, in which we focus mainly on solving problems related to post-harvest processing of sugar crops, such as sugar beet and sugarcane. So this is just a short overview of what I'm going to share with you all today. I'll talk to you about my own background in education and how I came to be here at ARS. And then I have a slide to share some different research roles present here at ARS in our building and throughout the agency, and what types of education levels and experience may be required, just to kind of give you some ideas as far as research goals or education goals, career goals, those types of things, and what kind of income you might want to make someday. And then I'll share some of my typical day activities in the office, in the lab. And then I'll just give you some snapshots into my research in the microbiology area and how we support sugar crop processing and some of the microbiology related problems that we work on. So just some random snapshots, nothing super comprehensive. We don't really have time for that today. And a lot of the work we do is multidisciplinary. So it's not just microbiology. It's also being willing to learn new techniques and collaborate with people in different areas. So we're always learning how to do new things to solve our problems as well. And then just, to wrap up, some other things that I do typically as part of my role here. On the right is a really cool picture of biofilm in a sugar beet factory. We actually took samples from this and obtained isolates. And so we deal with things from the factories that, are maybe stinky, slimy, unsightly, And then we turn them into really refined, nice research projects in the lab to hopefully solve problems for our stakeholders. But it's a really nice workflow. So just a little bit about me. I'm a farm kid from rural Montana. And just to clarify, I went to Montana State University in Bozeman for a bachelor's in microbiology. They have a really great program there with lots of hands-on laboratories for microbiology, not just general, but also medical bacteriology, immunology, mycology, things that are still serving me to this day. So I can't overstate the importance of these hands-on laboratory classes for real world skills. Um, and then as a 22-year-old, I moved down to New Orleans for grad school at Tulane University School of Medicine, where I obtained a microbiology and immunology PhD. And then from there, I did 3 different postdoctoral fellowships in research, looking at, molecular genetics with different yeast and fungal pathogens, that sort of thing. And then long story short, I eventually got hired as a research microbiologist here at USDA. And I've been here for probably around 5 years now. As far as various roles and education levels, I don't know if you have your cell phone nearby, you probably do, but this would be the slide I suggest you snap a picture of. These are some of the different roles and education levels that you might want to think about. All of these are ARS positions here, and they're always listed on usadjobs.gov. And I'm going to take you through a little bit of this. Long story short on this, we still have a hiring freeze, but I mean, they could open up anytime. We don't really know. There's a backdoor way to get positions here through ORISE appointments. They can be at the technician or postdoctoral level. These are always listed for various agencies across the country on zintellect.org. So I highly suggest looking through this website where you need to have your resume, your CV, your transcripts, letters of recommendation, those kinds of things. But this would be your best bet at this point in time. Just in general, people can get hired here with LA appointments or ORISE with an associate's or a bachelor's degree. A lot of times these are university students or someone who's just graduated who can be hired at GS4. Sometimes it's for summer field work. There are different technician levels you can get where you need usually at least a bachelor's or a master's degree. So that's something to think about, entry level for some of these nice technician jobs. And then the next level is support scientist that pays at GS 9 to 11. Usually need a master's or a PhD for these roles. Although I know one exceptional person with a bachelor's degree who was hired as a support scientist. She is really good. And then postdoctoral fellows, GS 11 to 12, you need a PhD for these. And then SY, principal investigator research, X, Y, and Z. This is my position. And these pay anywhere from GS 12 to 15 or even super grade, depending on, where you're at in your career. And this requires a PhD and usually postdoc experience as well. So, it's helpful to think about what kind of role or position you might want to have if you want to do research someday, and think about what kind of income you might want to have someday. And you can always look up these different GS levels that are state specific online, just to, think about your future and those types of things. But again, these are the main websites you want to check for these types of opportunities. All right, so just typical activities for my day at work. Do my emails, I drink my coffee, and then I always check in with my staff. I have 2 technicians and a postdoc right now. I help coordinate the research activities among the team members, see who needs help on something. I'm involved with planning and setting up experiments. I spend a lot of time going over data curation and data management now that we have more people and we have a lot of projects. So I'm always checking over data, making sure it looks good, checking for mistakes, and then managing, ensuring we keep track of our data and where it goes. And so this is kind of an evolving thing that we have to get better and better at. And it's kind of even becomes a little bit of a problem. The more data you have, the more people and projects you have, which is really, really great, but you really have to keep track of it and do a good job organizing and maintaining it. And also training your people with basic skills like saving files with good file names and not saving over other people's work. So these are basic things that, I have to train people on in my lab. We also do almost weekly lab meetings for people to present their research. And, for me to get everyone on the same page so I'm not giving instructions separately to all the different people, and it kind of helps streamline things. And I'm just in the lab. My office is within the lab, so I have my lab radar on and I use my senses, my eyes. Does everything look right? I'm listening to see if there are noises that machines shouldn't be making. I'm smelling to see if anything's burning, those kinds of things. Just being really aware and being a good steward of our resources and our instruments. And these are things that I also try and train my technicians, especially to use their senses, to be like a lab mom and make sure that nothing's amiss, nothing's going to break, nothing's catching fire, these kinds of things. And then when I'm not going through the lab and checking on people, I just spend most of my time writing manuscripts, reviewing things, doing proofs, submitting things for publication, responding to reviewers. I write a couple grant proposals every year to support the extra people in my lab. So that's always an ongoing thing, as well as, annual reports, progress reports for different projects and funded things that we have. And then I also perform peer review for a journal published by ASM called Microbiology Spectrum, as well as some other journals. And this is a nice way to give back academically and support the scientific process. And I also do it because I always learn something. I learn something new when I perform peer review for others. And then another thing that sometimes we don't emphasize with this work balance and performing well, especially as working women, is taking care of ourselves. And for that, I try and eat healthy. I try and exercise regularly. And we're really lucky that we have exercise facilities on site. And I find that when I take care of myself and I exercise regularly, my performance goes through the roof. And it really supports not just physical, but also that emotional, mental, cognitive balance and performance, because we know that stress and multitasking and managing people can really take its toll with stress in our lives. So I really promote that as well and try and be a decent example of work-life balance. And then the next thing is I have to work efficiently and use my time wisely when I am at work, because I have to leave on time to go pick up schools for my kids from school at their aftercare and all of their programs. So a lot of it is time management and disciplining myself to get things done within my time that I have allotted. And this is something that I also try and instill in the people in my lab. Transitioning now just to tell you a little bit about the sugar industry and then how my research kind of supports the sugar industry. If you look at the map of the United States, up north we have the sugar beet crop that's grown across some of these northern states, and this accounts for about 55% of domestic sugar production. And then down south, mostly in Louisiana and Florida, we have sugar cane grown, and this accounts for about 45% of domestic sugar production. But in all, the sugar cane and the sugar beet, each of those industries are roughly $4 to $5 billion industries each per year. So it's a significant part of the US economy, and it also supports our food supply here in the country. So how does my research relate to sugar crops? Well, if you look at this beautiful picture of a sugar cane field here, this is one of the crops that we support their farmers and factory operators. And so when these are harvested out in the field, they get cut into these short, maybe 9-inch billets. And it's a similar process for sugar beets. They get harvested and trucked in and stored as piles up north. Well, after they're harvested, there can be injuries to the sugar beets. And also where the cane is cut, it gives access to all of these microbes present on the plant and in the soils. And once the microbes gain entry to the sugar-rich environment of the plant, they use it as a carbon source to grow. And so they consume the sucrose. And a lot of the microbial load and composition will vary across different fields with the amount of soil, There are all kinds of different variables that can affect this. But moving on, just to tell you a little bit more, we think of these as microbial communities. We've used different types of techniques to study this, culture dependent, culture independent. And then from there, we've done additional testing, such as susceptibility testing. I'm going to show you a few pictures for that to determine optimal concentration levels of antimicrobials to stop microbial growth. We've also done genomic and bioinformatic analysis to identify key genes in some of our isolates. We've also done some carbohydrate chemistry study to study bacterial exopolysaccharides. Why should we study EPS? These are long sticky sugar polymers, also referred to as gums, or if you're one of my kids, they call them sugar boogers because that's what they look like. These are key structural components that play a role in biofilm formation on factory surfaces. And they also cause a lot of operational problems in the factories that interfere with the raw sugar extraction process. These EPS are often either glucose polymers or fructose polymers, and this just highlights further the multidisciplinary approach to our research, that it's not just microbiology, it's also having a good knowledge of chemistry, a good knowledge of, genomics, of DNA, of all these different areas that work together in our science. I'm speeding through a few of these slides a little bit because I want to get through in time for you guys. But as I mentioned, we have done a lot of genomics and bioinformatics. Just in the last year, we've sequenced and assembled about 38 bacterial genomes. We've done species level identification through various pipelines. And we've also used a protein annotation pipeline at NCBI to identify key genes of interest that form some of these EPS polymers that we're interested in. This is an example of some of the isolates that we identified and isolated from various factory samples in the past few years. We just published these genomes at an ASM Journal of Microbiology resource announcement so that all of these genomes are publicly available to the research community on NCBI. As I mentioned, we also have done a lot of susceptibility testing, and we continue to do this by microdilution assay. All of these are performed in a biosafety cabinet type 2 so that our samples and experiments are all protected from contamination, hopefully with proper aseptic technique. And that also protects the user, in this case, Zainab here, from acquiring any infection accidentally from any of our samples, especially when they're unknown microbes that we haven't identified yet. So this is Zainab. She came through the biotechnology program there a few years ago, and so she's been working really hard and doing a lot of work for us in the lab here. Here's an example of some of the data that's been generated from the susceptibility assays where we determine minimum inhibitory concentration values, meaning the lowest concentration required of a particular antimicrobial agent to stop microbial growth in our growth assay. So here's just an example of some of the isolates that we've tested, the resulting MIC values for each of these antimicrobial agents. Million or ppm is what the industry uses. And then you should note that each of these numbers listed here are the result of at least 3 independent experiments. So each of these data points is part of a collection of hundreds of experiments to generate these data. And so it's really robust, and the workload should not be underestimated. These are all of the different values. The lower values mean that antimicrobial work is better than the things that have really high numbers. That means that they work much less. They're much less efficient at killing and are, probably not a good idea in the first place. And I should say, I don't really have time to go over a lot of this today, but it's in press and it will be available with open access. All of the papers I'm telling you about today, and I can always send these to you. What else? We also have a project growing and modeling and studying biofilm formation on factory surfaces. And so for this, we have these really nice biofilm bioreactors with individual growth chambers here. This is depicting the flow through phase where we drip media over these coupons to look at biofilm accumulation. And so we had a paper just published on this. But this is what it looks like when we scrape the biofilms into water for homogenizing and measuring CFUs. We do all of this in a biosafety cabinet when we open these up, especially when we do homogenizing, it can create aerosols. And so we have to follow proper safety procedures. To quantify and measure our CFUs, we're really lucky to have this instrument called a spiral plater that you can select different programs to plate different volumes and different patterns. And then so your samples are plated onto these auger plates, and then we can image them with a special imager. And then we have a special software that counts these colonies. So this really helps to streamline and improve accuracy of our and makes it easier to get CFUs per mil counted on several samples and run our duplicates or triplicates and get some really nice data. And so this is what the data looks like when it comes out in terms of average CFU per square centimeter on these coupons. Again, this is available online if anyone wants to look further. It looks like I'm a little short on time. We also take our biofilms and we also are able to image them by cryo-SEM. And so this is at 5,000 magnification showing phenotypic variation in the cell density and some of the structures that they form. So shoot me an email if you like a copy of this paper or the link. This is pretty cool. It just came out. And then just things in a nutshell. My position is a dedicated research position, so I'm not required to teach. We have appropriated funding from Congress, which is really nice, although I do have some extramural funding from various stakeholders. I do supervision. I interact with stakeholders a lot and try and get their input because we're trying to solve problems for stakeholders in the industry. Lots of writing and data management. I'm also a biosafety officer. These are the folks in my lab right now. We have Sanjay, the postdoc, and Tia and Zainab, who are technicians. Here are two of them working in our brand new biosafety cabinet that accommodates 2 people now because it's 5 feet wide. So we're very excited about that. And if you have any questions, I'd be happy to answer. You can always shoot me an email. Is there anything I can answer for you all?
I have a question, and I'll make it really quick just because I know we are short on time. What are specific skill sets you would want students or graduates to have if you were able to hire at this time? What do you look for? I know you mentioned some of it before, but.
Okay, yeah, pipetting and basic culturing and aseptic technique is really crucial. But something also is filling out Excel sheets, being able to basically use Excel. Basic skills like properly naming a file so that you can come back to it and figure out what it is. Organizing data. I think attention to detail, making sure that you crunch your numbers correctly and just learning how to learn and how to check yourself because it seems like there's always things that can go wrong in experiments and you have to be able to have good notes and go back and kind of figure out what's going on. So good attention to detail, being organized, taking good notes and writing good lab notebooks that you can go back and read your notes or so that your supervisor can go back and read your notes. And some people are really, really good at this and other people take really good notes and my brain is just glazed over and I can't read anything. So those are some practical skills I would say.
Will you just repeat lab notebooks one more time?
Lab notebook is huge. Not that you have to have perfect handwriting, but it has to be organized and spaced out with headings and dates. Otherwise, it's not of use to anyone. And we even put little notes in there, on our Excel sheets. This is on page, 90 of notebook 3 or notebook. In our notebook, we might say saved as this Excel file because 6 months or a year from now, we'll be going over our data and writing a manuscript. And if we can't find it, it's panic attack central here because where is it? Didn't we do that? So those are just some basic things that make life better for everyone. once you're cranking out data.
Thank you. Everything you said is stuff we really emphasize to our students. So I'm glad you brought up some of those soft skills, you know.
Thank you so much. We appreciate your time and your support of the program and Delgado. And if anyone does have any questions, please email. You can email myself, Ms. Shunik, or Dr. Bruni to ensure that you get it answered. Again, we appreciate you, and we're going to head right into the next speaker. I'm going to do a quick introduction. Let me share my screen again real quickly. Okay. Our next speakers are a duo. I'm not sure if Miss Robertson has been able to hop on yet, but I do see that we have Hubert Franklin on. So Hubert Franklin is a Louisiana native and the project leader for Veolia North America's New Orleans project. Devoted to water and wastewater industry, he supported municipalities, I cannot say that word, I apologize, statewide, and trained more than 500 operators for certification. He advanced to operation manager for Veolia's Harahan and Kenner sites, later becoming assistant director of sewage for Kenner, and he's then served as sewage director for Jeff Parish. And he's overseeing Louisiana's largest collection system, including 5 wastewater treatment plants and over 500 lift stations. Hubert, is Nadine on? Did I miss her?
Yes, I'm on. How
Oh,
y'all
yeah.
doing?
Hey, Nadine, let me do your introduction also. I'm sorry, I didn't see you hop in. Okay, Nadine Robertson is a seasoned wastewater professional and entrepreneur based in Greater New Orleans region. As a Class 4 wastewater treatment operator and WWTP project manager, she brings extensive experience in wastewater operations, industrial maintenance, and environmental compliance. We welcome y'all. And again, if anyone has questions, you can put them in the chat or you can wait till the end and ask them directly. And let me stop sharing and y'all can share the screen if you need to.
Okay.
I just gave you permission, Nadine. I'm sorry about that.
Gotcha, gotcha. Okay, can you guys see my slide deck?
We can.
Awesome, awesome, awesome. Alrighty, Nadine Robertson, as Amanda stated, I'm a project manager, project leader for Veolia North America. I'm actually operating out of Kent, Mississippi, which is about 30 minutes outside of city of Jackson, but I'm a native of the Greater New Orleans area, grew up in Harvey on the West Bank. So just a quick summary about Veolia. We've been around for 170 years. We're operating out of 44 countries, and we run about 300 plus projects in the United States. So we provide O&M operations and maintenance to a lot of municipalities, a lot of people in the private sector, overseeing the day-to-day operations of wastewater treatment plants as well as drinking water treatment facilities. We also provide consulting, engineering, and a whole bunch of other services to the clients. Some of Veolia professional development opportunities, we participate in WEFTEC, and I'll discuss what WEFTEC is in a couple of the later slides. But the Water Environment Federation hosts a operations challenge every year at their WEFTEC conference. So we have a picture of the NOLA mud bugs participating locally in the operations challenge. Veolia also affords a lot of internal development opportunities, such as the Women in Leadership program, which allows us female participants to get together and network and discuss some of the issues we faced as women in the industry. As you guys know, it's kind of male dominated. We also have an operator certification committee Well, we get together and discuss different bonuses for the operators to maintain their CU hours as well as their licenses within a company. Discuss some of the different test questions and things like that, which the operators face for the state tests to obtain their licenses 1 through 4. Veolia also affords a lot of tuition reimbursement. So if you want to go back to school and get a degree or take any sort of certification courses, we do reimburse our employees on that. Veolia also supports us in various ways to attend conferences such as WEFTEC, AWWA's conference, which is ACE, statewide tests such as statewide conferences, like the Louisiana Rural Water Association conference, various community service projects we participate in, such as Waterpalooza, which is an elementary science school community fair that WEF puts on during WEFTEC week. Various travel opportunities where we can go and get hands-on experience at our sister sites, as well as J.J. Kellers and various safety credentials that we do in-house, ranging from first aid, CPR, forklift certification, CDL licenses, and crane operators. This a quick bit about myself. Like I said, I'm a native of New Orleans from the West Bank. I got my start in 2018. I started off actually under Hubert when he was the operations manager at the city of Kenner as a belt press operator. Later transitioned over to the lower Ninth Ward plant, which Veolia provides operations and maintenance for, and Hubert's also the project manager over there. He could talk a little bit about himself and that project. From there, I kind of transitioned to Greenville, Mississippi to oversee a plant through a company called Manshack, which is a civil engineering service company. I was there for 2 years. Then I came back 2 years to date back to Veolia to oversee a wastewater treatment plant in Canton, Mississippi, where I'm currently based at. This particular slide, I just wanted to highlight some of the certifications and some of the higher education degrees that you can take. I consider myself to be a lifetime learner. Some of these certifications range from OSHA 30, HAZWOP for 40 hours. NCCER is a national construction certification. They offer certain specialty credentials in pipe fitting, electrical, maintenance, safety, whole bunch. And I'm going to pass it over to my counterpart, Hubert, so he can discuss his career journey.
How you guys doing? Just to share some insight about myself and what was mentioned earlier, I'm a biochemist and I'm the project leader for Veolia Water. I oversee the biggest wastewater treatment plant in the state of Louisiana. We have a treatment capacity of 120 MGD at the East Bank and a 15 MGD trickling filter plant on the West Bank. Throughout my career, I was able to work at 11 different wastewater treatment plants. Just like some of you going through college, I remember taking classes at Delgado, some of my organic chemistries and general chemistry while I came down for the summer. But this is a field that's intricate and is very science-based, but we also have some maintenance aspect that goes along with it. I was able to train well over 500 to maybe 800 operators in the state of Louisiana. I'm an LDH approved operator. And it's my job to cultivate the microscopic bacteria to treat the incoming BOD that you guys flush down the toilet on a daily basis. So I just want to share a lot of knowledge and let you guys know it's important to flush the proper things down the toilet. So that makes our treatment that much more easier on our side. We cultivate 5 microscopic bacteria. That's going to be the amoebas, the flagellates, the cellulates, the stalk cellulates, and the rotifers. We cultivate those bugs so we can chew up that trash that you guys send to us. So like Nadine mentioned, I think we love our career. We love the journey. I've been in for 15 years. I was able to take over for Baton Rouge, Jefferson Parish, where I had the biggest collection system in the state of Louisiana. I had to oversee 5 plants and 520 lift stations, 1,400 miles of gravity enforced main piping, 22,000 manholes, and I had to operate a $78 million budget. So I started out as an operator painting and cutting grass. And within 15 years, I worked my way all the way up to the top. So just with a little will and determination, you can just to show that you can become whatever you desire to be. So that's what I wanted to share on my behalf. And Nadine, you can continue on with your presentation.
Okay. So I briefly wanted to touch on two of the leading associations in our industry. One of those being AWWA, the American Water Works Association, which focuses more on the drinking water side of our industry. We also have the Water Environment Federation, also known as WEF. This is the organization that hosts the WEFTEC, Water Environment Federation Technology Conference. It's based in New Orleans, and they rotate between Chicago and New Orleans every other year. It is a very big deal conference, allows the opportunity to network with students and young professionals, which is kind of how I got acquainted with the organization, as well as senior and executive level professionals in the industry. So their Students and Young Professional Committee, also called SYPC, has a wide range of opportunities where you can get involved with, such as the EYPL, Emerging Young Professional Leadership Conference. Upcoming this month later on, they're going to have the Utility Management Conference, which is partnered with AWWA and WEF, where they have a summit specifically focused on young professionals and, how to ask for a raise in your career, some of the skill sets you should have to be successful in the industry, and a lot of other topics that college students and high school students would benefit from. They also have a student design competition, which is for the college students, where they present a research topic at WEFTEC for scholarship dollars. WEFTEC Waterpalooza, like I said before, is an elementary science fair community service project that takes place during the week of WEF at a local host elementary school. They have a mentorship program where they match students and young professionals with senior and executive level professionals that's in the industry. And they also have what's called WEF Max. So each state has a member association. And this is the opportunity where each state member association can get together and kind of work on best practices on recruiting new members, how to put certain events that have been successful in other MAs, and how they can reproduce that. WEF also has a program called Info Romeo, which is called, that's my dog, I'm sorry, I'm at home, called Introducing Future Leaders to Opportunities in Water. This particular program is focused on minority HBCUs to incorporate a lot of more minorities into the water industry. There's two different tracks. One is called STEM, which focuses more on those seeking undergraduate and graduate degree programs. The second track focuses on non-traditional students, so people that's doing a career change or people that's new to industry that are not seeking a degree. So that's the difference between the two, and they are also recruiting right now. I believe I have a link to the application and the last slide of resources, in which your teacher is also going to send out this slide deck. So feel free, if you're interested, to go ahead and take advantage of this opportunity and apply. These are some other national associations that are essential to our industry outside of AWWA and WEF. WPI was formerly known as ABC, the Association of Board of Certification. They are focusing on standardizing these operator certification exams. I believe there's 6 or 7 states that's under WPI at this point. NSBE is focused on Black engineers. SYE, Society of Women Engineers, is just women that's focused primarily on becoming engineers because it's very rare in our industry. SWANA focuses on solid waste. So when you're dealing with, our belt press operations, all of that cake and all of those solids that either go to the landfill or lagoons or whatever, they focus on the solid waste, the biosolids from our wastewater treatment process. And touching briefly on Operators Without Borders, this is a volunteer-based organization to where they send certified operators to third world countries to help create better quality life and water for those third world industries. Some of the benefits of joining these associations include obviously scholarship dollars for continuing education. You gain leadership opportunities and volunteer on committees such as SYPC to where you can increase your network. I had the opportunity to participate in a fly-in where I was able to go to DC and advocate against these so-called non-flushable wipes, which cause a lot of clogging issues at the wastewater treatment plant. So we were successful in requiring these companies who make these wipes to put labels on their packaging that says, you know, they're not flushable. They should not be flushed in a toilet. So that's one of the big issues we've been dealing with outside of PFAS in our industry. These scholarship opportunities also give you the opportunity to travel to these different conferences. They pay for room and board. They give you book vouchers. So WEF has a ton of manuals as well as AWWA that you guys can take advantage of and help prepare you to take these state operator certification exams. Hubert, would you like to talk about some of the vast careers opportunities that's in water?
Yes, here in New Orleans, I mean, we have opportunities on the maintenance side. We have data analysts, we have lab techs, we have operators, we have construction management, upper management from accounting, purchasing. Those are the many different avenues that you can kind of focus in on to try to get into our field on the wastewater side. So I guess if y'all got any other
Just
questions.
also touch basing on that outside of the trades, you know, this is a business. So we have administration as well. We have to have our accountants, our executive team to keep the business in line, right? Outside of operators and maintenance, I would say also another essential part of that would be the instrumentation teams because we have flow meters and SCADA systems and all of that to have the operators maintain and watch in the control room because you have different temperatures, like at the East Bank plant with this incinerator that has to have a certain pressure, has to be maintained, temperatures. Also out in the field, you know, we test for pH, dissolved oxygen and things of that nature. Total chlorine residual on our outfall or our influent water. All of these different parameters have to be monitored, and we have a system called SCADA, so we have to have the technology behind that, and obviously someone has to be maintaining those upgrades and repairing if the computers or the software system has issues. Okay. Some of the skill sets that you would need to be successful in this industry, obviously outside of reading and basic math, just common sense stuff as far as coming to work on time, being organized, communicating, because there is a team, different departments that you have to interact with. Obviously troubleshooting, because you know every day is not the same. Certain issues may arise, so you have to know your process and be able to troubleshoot to know how to handle different problems and think out the box and be innovative. Sometimes you have to create solutions to these problems. Obviously, be safety conscious. Safety is the most important because, I mean, if no one's working safe, then everything else just goes down the drain, right? Hubert, are there any other skill sets that you think are essential to our industry that you would like to touch on?
Outside of what you mentioned, like I just said, pretty much basic mechanical skills. I mean, you might come to a point where even though you might not be a maintenance person, but you might have to break down a pump, and not to the extreme of being a professional maintenance guy, but just the basic maintenance, knowing your different types of tools and equipment, proper PPE understanding, protecting yourself, like you mentioned, for safety. But that's the basics of this a foul field.
Okay.
Excuse me, guys. There's a question for Nadine in the chat.
Okay. See if I can pull it up. Sharing this.
Nadine,
Can someone read
sorry.
that question? Because I'm sharing.
It was the same question I was going to ask at the end. It says, Charlene Varnado said, I'm interested in becoming a water operator. What do you recommend as the first steps for a career change? So this aligns with what I was going to ask. We offer chemical technology, biotechnology, and wastewater and water certificates at Delgado. We have levels 1 through 4 at Delgado in 4 different categories, which I saw were listed on all of your flow chart for ways to get into certain fields. Do you think it's best for students to do the associates with chemical technology or go the waste and water and wastewater certification route?
I would say the wastewater certification route. We're having issues in our industry with obtaining and recruiting certified operators. So if they're able to get a foot in the door and get, acquainted with that material and have those licenses before they even hit the ground, I think that would be beneficial. We do offer on-the-job training. So once they get, into the actual job, we're going to train them on everything they need to know. But those certifications are essential, I would say.
Thank you. Charlene, I'm going to send you an email later that just tells you about our water and wastewater program.
Okay. Veolia has recently partnered with WEF and AWWA to host a Veolia Workforce Academy, which is a free online platform where anyone can go on and get more knowledge base about treating wastewater and drinking water. Is 100% free. So you can go on at any time. It's very flexible. To get on here and go through these modules, you do have to take quizzes at the end of each module and pass with about 70%. At the end of each module, you will be able to print out a certificate, which you guys can keep in your portfolio when you go on job interviews, as well as submit to the state for CEU hours to test as well as maintain your licenses, right? So this is groundbreaking for our industry because most of these 32-hour classes, they charge about maybe $400 a head for CEU hours to take these classes. So this is free and you can do it in the comfort of your home, cell phone, tablet, or at work during your downtime. So we're trying to encourage more people to get into our industry because we're facing a great tsunami and we're trying to be more digitally flexible and more environmentally friendly by, offering this online free of charge. And that is it for the presentation. If you guys have any Q&A for myself or Hubert, feel free to ask away. These are some resources. So I'm gonna stop sharing my screen at this point so I can see you guys. All right. Any questions for myself, Hubert? And we also have our community engagement / business development person on the call, DeShannon Cobb. So if you have any questions related to her job and what a typical day looks like for her, she can also answer some of those questions. It's quiet group here.
I want to thank you. You answered so many things. We are very fortunate that we have Dr. Noble to help support the water wastewater program that we offer with the certifications. So I'm excited to share your slides to the attendees as well as information about that program.
Appreciate that. I mean, that's what we're here for. Hubert and myself were once a college student as well as DeShannon, so we were in you guys' shoes at some point. I actually did a career change, and so I started from the bottom, just as Hubert stated, and you get what you put into the industry. So, I mean, this industry is so vast, it's very lucrative, so you don't have to worry about getting laid off like some of the oil and gas industries and the refineries. So it's essential, it's public health, and it's going to be around forever, literally.
And Dr. Rosenzweig, I would like to share Nadine and Hubert are both trainers, operator trainers. So they train as well. Hubert trains for Louisiana Department of Health, and Nadine does as well. And I think that they can come to sites and work with other organizations to allow training, because we have the partnership with Delgado, with Dr. Noble and the workforce development team. But we also have these two awesome individuals that work for Veolia that trains as well for the entire state of Louisiana. So you have some great knowledge on the on screen here.
We appreciate everything. Thank you so much. And hopefully this helps us improve the communication and pipeline into water and wastewater.
Yes, ma'am. Thank you guys for having us.
Thank you very much. And I look forward to continuing our partnership.
Thank you.
So this leads us directly into our next presenter. I'm gonna grab the screen and do a quick introduction. Our next presenter is Mark Hiatt. He has his master's degree and 19 years of experience. He specializes in analyzing biological and chemical specimens and producing high quality scientific reports. He has a strong foundation in laboratory practices and quality assurance, and he excels in troubleshooting and maintaining instrumentation. I can vouch for that because he helps us almost on a weekly basis with one of our $500,000 machines, Mass Spec. And he currently serves as the Deputy Director of the Crime Lab for New Orleans Police Department. And he has previously served as the Lab Director for the Regional Crime Lab in Central Ohio. And with all of this, he brings extensive leadership and operational expertise to forensic science. So without any further ado, welcome Mark Hiatt. And let me stop sharing so you can share if you need to.
Great, thank you. Can you hear me?
Yes, we can.
Okay, perfect. Okay. So good afternoon, everyone. I was honored to receive an invitation to present from Dr. Rosenzweig about my career in forensic science. It's an awesome opportunity to sit in on this open house. So let me get this started. So I'm here to talk about forensic science, which is very much out in the TV world, but I'll try and give you some real snapshots on what lab life is like. That's my point of contact email. So if you ever, I believe Dr. Rosenzweig has it also. So
155 is not that big.
Yeah, so put those notes down. Here's what I'm trying to talk through today and the discussion point. It's just my journey into forensic science, what services we currently have at the NOPD Crime Lab, other forensic disciplines, and some ideas to get into the forensic job community. So I started in 1998 with my career. I walked into Malone College, bio pre-med. I quickly realized that med school was not the career path I wanted in my future. And I started looking around at what my skills were when I was in class. I actually switched majors my sophomore year and found my home in the chemistry program. And I got really good at lab sciences, right? Like labs are what kept me going. And then, so in 2002, I graduated with a bachelor's degree in chemistry. That concept that the prior speaker was talking about, that lifelong learning, is, I think, something that scientists are actually really good at in general. So I kept pursuing additional education, and the University of Florida has a great online forensics program. So I started that in 2006 and completed that in 2008, and it's under their School of Pharmacy, but it's a master's degree in forensic drug chemistry. They have programs that also focus on toxicology, biology. They have somewhat of a general master's if you're kind of getting the master's to get into the forensic community and you kind of don't know what discipline you're going to do. As my work career progressed, I started taking on supervisory roles. I was hungry also to get some of those leadership skills and that background, and then I found a great program through the LSU Shreveport. It was an online master's degree in business administration with a lot of my classes being leadership focused, some operational, some accounting, which I hated. I hated accounting, but as in any degree program, sometimes you just persevere, and really enjoyed that and all the things I learned. So doing the work, I started my career, graduated Malone, knew I didn't want to pursue any additional education at that point, and I got hired on at Abbott Nutrition. And if you don't know Abbott Nutrition's product lines, it's Similac, it's Ensure, it's a lot of medical nutritionals. Abbott also has pharmaceuticals, they have diagnostic testing. It's a multinational company. I was lucky and blessed to get in the vitamin lab doing HPLC work and learned a lot of great lab skills from an amazing array of analytical lab professionals. Still had passion to want to get into forensics and was able to onboard and get attached to the State Highway Patrol Crime Lab, which is also in Columbus, and started as a drug chemist and then transitioned into leadership there. I went back to Abbott for a year, and it was a good transition out. I learned kind of the manufacturing side and how that works in a chemical with my chemical background. Through a news article, I learned that, and Dr. Rosenzweig touched on that in my introduction, that the Licking County Sheriff's Office, which is just east of Columbus, wanted to open up a lab, right, and start from scratch, which when I say open up, I mean start from scratch. My lab that they were going to put us in literally had a sink cabinet in the corner and nothing else. So I had to go through that process of understanding like how to buy supplies and lab benches and instruments and write policies. But it was a great training ground for me to grow as a professional. There were definitely days that were like, what did I get myself into? But I loved the work, and then we got accredited, and I think that lended itself to moving down into my current role. And you know, as the deputy director here in New Orleans, we're also in that build cycle where biology is trying to get up and running, and some of the other lab spaces, how do we grow each of those disciplines that we currently have? So let me talk briefly about what we see in the lab, and some of my photos didn't show up, but I'm a trained drug chemist, so a lot of what I nerd out on is all drug chemistry, and this is just some of the things that we see, right? It's crystal material, it's plant material, it's random tablets, it's baggies of samples, and we have to be really strong in our analytical chemistry skills to say, hey, I know color tests and I know a lot of presumptive tests, so that kind of gives me the roadmap to understand what sort of confirmation techniques I need. And by confirmation techniques, I'm talking about your gas chromatograph, mass spectrometer, things that allow us to then go into court and sit and say, hey, you know, to a jury, you know, I'm confident that this particular blue crystal material is, you know, crystal methamphetamine. Oh, there's the photos. So we also deal with like photos that are pharmaceutical in nature, but also there's a lot of fake clandestine tablets, which is the red M30s that you saw there. And this is, a lot of what we deal with is very It's either legal or it's very focused through accreditation. So what we test in drug chemistry is prescribed to us by what's written in the Louisiana revised statutes. And then, so that's drug chemistry. Other services that we offer, this is something external to the lab. So it's a lot of going out into the field and processing crime scenes. So your bottom right photo is actually a screen grab that I got of some of our staff members that they were on a major scene, I believe in the French Quarter area, where they're kind of getting set up and ready to put out those flags and take the photos of the evidence that they need to collect, like, you know, bullet fragments and a gun. And then some of your other photos is, this was once our former sergeant here at the crime lab, you know, also processing a scene. So the crime scene's very external focused. So in addition to drug chemistry, which is more lab work, we also have firearms examinations. So we do a lot of test fires to get those rounds and then have a comparison microscope. So it's a lot of using attention to detail and understanding that line of like, is it an inconclusive test? Is it a match or is it not a match, right? So inconclusive in forensic world is, there are some similarities, but I'm not confident, right? And that's not a negative, it's giving benefit of the doubt to that defendant. We also have latent prints in the lab. So this is those fingerprint comparisons, both in development and in comparison. So sometimes the friction-rich unit will use just hand magnifying glasses, but we also have our APHIS station that uploads things into the national database. We're working diligently to get our forensic biology unit up. The city of New Orleans hasn't had biology as a testing option since Katrina. So city council, I think around 2022, had this big push of like, hey, we're spending a lot of money outsourcing. What does it look like for this discipline to come back to the city of New Orleans? And in your bottom right photo is actually two of our analysts in the lab and two of the instruments that we use, them doing some validation work. But for biology, you use those alternative light sources to figure out where to do your best cuttings out of, let's say, a cloth or a bed sheet or a pair of underwear, and other processing techniques to eventually get to the point where you're uploading that into an instrument. And then these are some of the other forensic disciplines, right? There's toxicology, there's digital forensics, because everyone's carrying a computer around with them, right, in the form of their phones. Anthropology, forensic nursing, you have forensic pathology, which typically for those roles, which forensic pathology is very much a need in our community, you have to go through that process of getting into medical school. Question documents and trace is still used. It's used a little bit less, but there are still some skilled practitioners in that. And then arson and fire analysis, which is a lot of your similar skills that drug chemists would use for that GC work, that gas chromatograph. Just some getting into the role requirements. We, as a lab, and most labs, go through an accreditation process, which it's an international set of guidelines in addition to some of the directives that we get from ANAB, our accreditation management, and say, hey, for these roles, these degrees are required. And typically it's usually phrased in a bachelor's degree in a natural science. So whether it's forensic chemistry, physics, chemistry, biology, some sort of natural science. And then some of your associate's degrees roles are latent prints, firearms, crime scene. Sometimes people just go through and get bachelor's degrees for those roles. And then sometimes you have officers that retire early and then step into a lab space. So it is a dynamic space, and the degrees usually, as you're looking for jobs, it'll list them. So that how people list them is a good guideline. As far as other advice, and I really think in this space, you have to be a lifetime learner. Like so many, as I've trained staff, so much of when I started in drug chemistry in 2006, it was very straightforward with what we were receiving. And it has just gotten weirder and weirder and weirder. So when you possibly hear a news story about a fentanyl, but now it's not fentanyl, it's acetylfentanyl or crotonal fentanyl or all of these different things that I thought I'd never have to use again once I graduated organic chemistry, I'm now using a lot more and more. But we're always in a constant cycle of being curious and learning. I mean, even one of our goals here at the lab is to constantly have those continuing education credits. The reason Dora the Explorer is on here, just from a jobs perspective, sometimes you get lucky and you go through your degree program and some lab has a forensic opening in the discipline you want and it's close to home, right? I feel like I got lucky in that regard, but there are many times, and I've had coworkers at other labs where she graduated from a university in Ohio and the first job that she was able to get was actually in New Mexico. And so she moved, and she was single at the time, so it was easy. She moved to Albuquerque and got on there, got trained up as a drug chemist, and then she kind of worked her way home. But other ways to get plugged into the community, and that's what the other photos are there's regional organizations that sometimes hold fall conferences or give the opportunity for student memberships, or if you're doing any sort of student research projects, they'll allow you to do poster presentations. And that's where you have professionals that could be hiring that go to these regional meetings and interact with you, and they might, grab your contact information. And so that's another way to kind of get yourself out and get yourself exposed into the forensic world. And recently, just in early February or mid-February, even the American Academy of Forensic Sciences was actually in New Orleans. And again, they had that same sort of like research project, poster presentation, come tell us what you did. And then you get a lot of great feedback too of like, other professionals that are curious about your research. Sometimes, I mean, the posters I went to at the American Academy, you almost want to take a photo and bring it back into the lab like, hey, can we implement this? So even some of those undergraduate researchers that are doing work, we can use it even after 23 years of analytical chemistry experience. It kind of, gets us curious here and is something that we can implement. But foundationally, and I think that's my last slide, what I would say is, and I sat in on 2 of the prior talks, when you're in that lab space, we really key in on having due diligence, attention to detail, writing things down. Like all of our notes are contemporaneous. So as I'm opening a case when I was a drug chemist, I received 1 sealed plastic bag containing 25 knotted plastic bags of white powder. And some of those language skills you get used to using both descriptive language, but you're also not using possible cocaine, right? We don't know that. We have to go through our testing process to be able to write that on the report. So our descriptive things that we use in reporting is very key. I think the other thing about forensic science, and sometimes, and this is something we typically ask in an interview space, is what is your comfort level in public speaking? Because yeah, you might be a rock star in the lab, but if you can't go into a jury where you don't know any of them, you don't know the judge, you don't know the defendant, you don't know the attorney, and you might only get maybe a half hour of pre-trial prep, you have to be able to go in, explain who you are, explain your educational background, what training programs you've done, how you've kept abreast of everything going on in the forensic community, and then relay all of your testing in the simplest manner so that you can say, yes, I am confident with all of my training, knowledge, skills, and abilities, in line with all of our standard operating procedures that I tested this correctly, and I'm confident that it is methamphetamine or cocaine or whatever the substance is. So I have loved and will always, I've always appreciated being in the forensic community. It just fits with the way my brain works, but I know for us here at the lab, we always love helping students get into this community. One thing I'll also advocate for too, because I know I'm getting short on time, is if you can find internships, that is helpful because that gives you that real world experience. So like if you get an internship and do a lot of drug chemistry work and you're like, this isn't for me, but firearms seems amazing, that helps you refine what jobs you apply for. And it also gives that lab that you interact with, like they might be hiring in 6 months. So if you go into that internship program and do really well, you're their first call. And I know, and I say that knowing we've done that. Like, hey, you were a really strong intern. We need you to apply for this job. And we let the process go through, right? If there's other stronger candidates with more experience, we take those, but at least, you have that opportunity to go in and interview. But yeah, I think that's it. And I can take any questions.
I have a question. I know we've talked about this before, but because some of my forensic science students are in here, can you inform the group about the minimum educational requirements to get into working at a lab and some of the other various roles within the forensic unit here?
Yeah, the educational requirements. So for drug chemistry, for example, because that's what I know the best, your education will need to be some sort of chemistry focus. So you're going to have to take like a Gen Chem process, at least one OChem, hopefully two, and some sort of instrumentation work. I think one thing, and Dr. Rosenzweig touched on it, Delgado has access to a very, very awesome piece of equipment. And I know it's still, like Tara Cullen, who's the lab manager, let's be honest, like she did all the work. I was just there to help. And she, that instrument getting up and running, like pouring yourself into that lab, whatever lab it's attached to, and Dr. Schuneck and Dr. Rosenzweig will let you know, and getting comfortable with some of those higher level instruments for drug chemistry and toxicology is very important, because that's what we do. We interact with them. A lot of times we do the maintenance on them in-house. And even when I was doing training for staff, we don't expect you to be a whiz at the engine. Like, we'll teach you on instrument maintenance. We don't expect you to be able to like tear it down completely and rebuild it. Not everyone does that. But we will get you to understand all of the nuances of the instrument and why things do what they do. But if you're the one that's like, I'll change the liner and that's it, I'm good. Because we want to give you that knowledge. It also helps to testify, right? Like if a defense attorney asks you some crazy instrument question, you, through our training program, have that knowledge base. As far as some of the other roles, and I know Delgado's more of an associate's focused, your avenues into this space is probably in crime scene or latent prints or friction ridge unit or firearms. And typically with those roles, we'll hire you in as a criminalist. And then as a criminalist, you'll start that training program. And then the plan is always, or would be, once you complete that training program, then we would try and find a role as a fully certified firearms examiner or latent print examiner. And the other part too, let me also advocate, as an associate's degree, sometimes you might, like me, you might have to go to a private lab and get the experience. And even when I was at Abbott, like I started at the University of Florida when I was at Abbott because I said, hey, this company, private company, is going to pay for me to go back to school and get my master's degree. So why not maximize that free money? And typically they want you to stay on for a little bit, but if you can't get right into a forensic labs, what's something forensic adjacent where you're learning the same skill set so that when you come in for that interview, you can speak on understanding standard operating procedures, understanding good maintenance, understanding good note taking, understanding just a lot of the things that if you walk in knowing those and speaking on those in your interview, we're going to look at you and rate you a lot higher than someone that just graduated from a degree program. And hopefully that answered your question.
Yeah, thank
I know,
you.
I think there's aspects of forensics in general, because even at like the American Society of Crime Lab Directors or the American Academy, there's a lot of people at degree programs that are trying to figure out, much like you at Delgado, how do we train up associates and bachelor's degree people to be as prepared as possible to walk into a forensics role.
Mark, there's one question in the chat. I know we're really short on time. And if you want to say we can email the answer, that's fine too. But how does a student apply for an internship with the crime lab?
That's probably an email question.
Okay.
Yeah, if you send that point of contact, I will be happy to respond. Because
Okay.
we did have a kind of a long pause on our internship program, but we're trying to ramp it back up.
Awesome. I will share it out with the individual that asked, and they can share it with the people that they're with. I appreciate your time, your commitment to Delgado, your support of Delgado and our students. Thank you so much. And I will see Mark Saturday, March 14th at STEAMfest. I know, Mark, you won't be there, but your colleagues will.
Yeah, Dr. Kelly will be there, and she's an amazing resource. If you want to nerd out on biology, she is the person. But we'll also have one of our biologists and a crime scene person there.
Thank you so much, Mark. We appreciate you. And that moves us to our next speaker. Our next speaker, is it sharing the screen? Charlie, can you see my screen?
It's still on Mark's slide, but
Oh,
yes.
okay. Okay, is it on my screen yet? It is. Okay, perfect. Our next speaker, very close to home, is Lauren, and I'm probably gonna get this wrong, Lauren Swientoniewski.
Swientoniewski.
Swientoniewski. Okay, awesome. Let me give you just a quick intro. Lauren is a postdoctoral research fellow with USDA, and she brings her expertise bringing expertise developed during her previous role as a research assistant at Oak Ridge National Laboratory in Tennessee. She earned her PhD in biomedical sciences from Tulane University School of Medicine and has a very strong background in ELISA, food allergies, and protein assays. In addition to her research career, Lauren serves as an instructor at Delgado, and this is where she supports our students as they navigate foundational biological science courses. So welcome, Lauren.
Hi.
And I will stop sharing so you can share.
Okay. All right, can everyone see it? All right, perfect. And everyone can hear me, right? We're good? Okay. All right, hello, I am Lauren Swientoniewski. I'm really thankful to be here today and tell you guys a little bit about me, how I stay sane as a scientist, how I got to where I am, and some career advice and a little bit of my current project at the USDA here in New Orleans. So a little bit about me outside of the lab. I know I can always work from home with my cat, Lana. I've lived in New Orleans for a little over 10 years now. I moved here from Tennessee. I love football. I love hanging out with my friends. I love running. And all these things help keep my work-life balance at a good spot so I can bring my best mind and research to work every day. So this is kind of my career journey. I am originally from Knoxville, Tennessee, and I graduated from the University of Tennessee in 2014. I got my bachelor's in biochemistry and cellular and molecular biology. I also minored in chemistry, where I took a lot of classes in analytical chemistry. There I got my main foundation in my chemistry and biology and biochemistry classes. At the time, I thought I really wanted to go to medical school. That was like my goal. And in high school, I was like, I'm going to medical school. And I went to a seminar about alternative careers in sciences, and I realized that just because I am good at science and biology and I enjoy it, medical school isn't the only option to apply my skills. And so I decided to join a lab, a research lab in undergraduate, at my undergraduate institution for credit. And I fell in love with actual like lab bench science versus medical clinical science. And in that research, I actually worked with plants. I guess because I was the undergraduate, I was the main person in the greenhouse, planting the plants, making sure they didn't die. I also did a lot of PCR and DNA purification and running gel electrophoresis for the different graduate students that were already in the lab. And it allowed me to write an undergraduate thesis and present at like their research days. And so that gave me a really good foundation in lab bench science and presenting my work. In between my undergraduate and my graduate degree, I actually was an ORISE intern at Oak Ridge National Laboratory in their mass spectrometry department. I was there for a little over a year, and my boss there made me present with his graduate students in their journal club and helped me with my applications to graduate school. And so mass spectrometry was a lot different than what I was doing in undergraduate. So I did that from 14 to 15. Then in 2015, I moved from Tennessee to New Orleans and started my PhD in biomedical sciences at Tulane University School of Medicine downtown. That program's set up where the first 2 years of your PhD, you're in a lot of different classes, and you rotate between different labs to find your home for the rest of your PhD. And I actually, during that process, wasn't sure what research I wanted to do. And so the rotations were really critical for me to decide my future career path. And I ended up with Dr. Diane Blake, and she actually had a grant in marine microbiology research. So definitely not in the medical or biomedical sciences field, but I still use a lot of protein assays and protein purification and growing cell cultures and bacteria in my graduate school time that can apply to what I do now. So there I did oil spill bioremediation, and I looked at marine microbes that naturally exist in the ocean that will degrade oil. And I looked at the differences in their growth when we applied different structures to their media. And I worked heavily with Tulane, the Uptown campus, with their chemical engineers with that. So I graduated in 2022, and actually I found my current PI or my boss at USDA, Dr. Sohaila Maliki, through my PhD because she worked with my PhD advisor. and I actually went to a lab meeting here at USDA years before I even applied for a job here, and they helped me write my preliminary exam, which is a part of your PhD. And so I joined this lab in 2022. Also, Dr. Jacqueline Nesbitt is a part of our lab. Many of you probably know her as well. And that's also through ORISE. And I'm doing my postdoc. I work with cross-reactivity, look in different nuts, like peanuts and tree nuts. And at the same time, starting last semester, I became an instructor at Delgado with their introductory biology lab courses. So, all right, so I'm going to go over briefly and I attempt succinctly at what my current project is. This is just one of many projects that are not only in Dr. Maliki's lab, but at the USDA ARS here in New Orleans as a whole. Because this center also does a lot of cotton research. They do a lot of food sensory research, looking at different flavors and tastes with different fish. I know that other scientists also look at toxins in a lot of produce and different plants. So here we have a couple of labs that do food allergy. So what is food allergy? Well, it's an immune response to a food that your body thinks is harmful. So I know I have food allergies. That's how I became interested in this field. I'm allergic to peanuts. And from that, my allergist told me to just generally avoid all nuts. And I have other food allergies such as shellfish and soy. But everyone probably knows at least one person that has food allergies. And so in America, there's over 15 million people among children and adults that have food allergies. And the prevalence of food allergies is steadily increasing. So when you have an allergy or when you have a reaction to a food, you can obtain this through eating the food, you can touch the food, or you can inhale the food. This also applies to other environmental allergens such as like tree pollen, grass, etc. But we're going to focus mainly on the food allergies. For me, I work with peanuts mainly and also cross-reactivity with tree nuts. This includes walnuts, pistachios, hazelnuts, nuts like those. So how does the immune response start? Well, it's all breaks down to the proteins involved. So different plants and foods have a bunch of allergens. Those are proteins. There's multiple allergens in a food, and your body decides, well, I don't like this certain allergen or multiple allergens within the food, and that will initiate an allergic response. For allergy, we're looking at, among the 5 types of immunoglobulins, we're looking at IgE in the middle. So in general, I'll go through this really quick, but when you are first exposed to an allergen, they are exposed to your antigen-presenting cells. They present it to your helper T cells, which stimulate the production of your B cells. And when you're in your second exposure to the allergen, your body has now developed a way to bind to the allergen that you were exposed to and initiate a allergic response. That includes histamine. So this can involve something called anaphylaxis, which means that multiple systems in your body have a reaction. You could get your throat starts to swell. You can start having a runny nose. Your eyes start to water. They become red. You can break out in a rash. And even in some cases, all of this accumulates to going to the hospital using an EpiPen and even potentially death. So this is, I'm going to try to break this down to be not as complicated, but a lot of peanut allergies also exist with other allergies, including tree nuts. This is called cross-reactivity. So your body went through the whole process of recognizing the allergen proteins, and they look similar to allergens in a different food, even completely different, like shellfish and peanut. And when your body's immune cells see these proteins, they think it's the allergen that you are allergic to, when it's actually in a different food, and that is called cross-reactivity. And cross-reactivity can be studied using different types of proteins and whether they are in their folded state or they are linear. So looking at the different peptides that are in the protein. And in the foods like peanuts and tree nuts, there are proteins called viscilins. And all you need to know about viscilins is that these peptides within these proteins have a distinct 3D structure. And we think that the IgE or our immune cells, they recognize not only the sequence of allergens that are in this protein, but actually the way that they are folded in your body. So they look at the shape, and that's what triggers your allergen or your cells to bind to that allergen, and you become allergic. So I'm going to highlight different molecular and immunological techniques that I personally use in the lab. I help collect and process donated blood that we get from either collaborators or sometimes from volunteers through Tulane. So if you have a peanut allergy and you want to donate blood, we are always accepting your blood, and I'll process it. I also use something called an immunocap Isaac test. This is looking at different proteins in their folded form on like a chip, and we apply the patient sera through their blood on this chip, and you get a readout of different allergens that you have binding to. We also use something called a peptide microarray, and I'll go into more details with all of these. This looks at linear or the straight sequence of your peptides. And then finally, something called an ELISA, which is called an enzyme-linked immunoassay. So I help process this, the blood. We use a whole process where we spin it down in a centrifuge, and we are able to separate the red and the white blood cells and the patient sera that we then apply to our experiments. I went over what the Isaac test is. This is a really cool glass chip. It's very small. On that chip, there's really, really tiny dots of triplicates of whole allergens or whole proteins of different allergens among different foods. And so we apply the patient sera to this chip, and we're able to see how your IgE, which is important for allergy and allergy response, how your IgE is binding to the different proteins or allergens on the chip. And I'm not going to get bogged down in the details here, but this is what an output from the Isaac test looks like when you export it from the instrument. This is just a couple of the allergens on the chip, but there are, I want to say, like over 100 of allergens, and they're all in triplicate. And so you get this really good colorimetric graph showing the different levels, with red being the highest binding, then orange, and then yellow, and then also 0 if it's very low. And so we use Isaac a lot of times to kind of hone in on certain patients that we have in the lab, what their Ig is binding to, and so we can use that to then take that sera and use it in more complicated experiments. Patient sera is a pretty precious resource that we have, especially if it comes from children, because children can't donate as much blood as an adult. And so we have to be really particular on patient sera that we have and what experiments we want to use it on because of the small amount that we have. So the peptide microarray, instead of looking at whole proteins, we have printed, it's once again a chip with very, very, very small printed 15 long amino acid sequence, and they're offset by 5 amino acids. I'll show you what this looks like. So that way, when we apply the patient sera to the chip, we can see how this IgE binds to the linear sequence. So it's not looking at the 3D or the conformational binding, but looking at what specific amino acids and peptides in the sequence the IgE is binding to. And we put this in an instrument, and it gives us, let's see, it gives us a readout of the different binding levels to the peptides. This is an example of what the overlapping peptides look like. So with the underlined, like that's our sequence of the 15 amino acids, and then it's offset by 5. So each time it bumps it up by 5 amino acids, and so we can look at the range throughout the molecule of the Ig binding. This is an example of what screening looks like. So I said there are a lot of really, really small dots, and we zoom in, and when I work from home, I'm able to have my trusty assistant, Kat, be very jealous of me working, but also wants to help out. So that's her on the right. But we do a lot of screening, and we get something like this, where another postdoc in our lab, Dr. Ian Rambo, he's our main computer guy. He can get the files from this process and be able to map it out and find different really hot peptides that different patients are binding to. And so this is an example of what this data looks like on the linear or the straight line level. Oh, and also we can use this to differentiate between different allergic statuses. So with our subjects, we have those allergic to just peanut, and we have those allergic to just walnut. And we also want to pick out subjects that are allergic to both of those allergies. And if we look at the output, we can see whether there's a difference between the different allergies that these patients have, and see if there is one that's overwhelmingly indicative of one or the other. Finally, the ELISA, or the enzyme-linked immunoassay, is one of the main experiments I do in the lab. I use this like in a 96 well plate shown on the bottom. It can go up to really, really tiny, like 384 well plates. Those are stressful days on my mind and my eyes. But essentially, you incubate your plate with allergens or proteins, and it sticks to the bottom, and you can add different mixtures of allergens and proteins, and it competes to binding to the bottom of the plate. And when you wash your solution away, you can use a spectrophotometer to essentially see competitive binding. And so whether your the sera or the antibody that you put in the plate, whether it's preferentially binding to one thing or the other in solution. And on the bottom right is an example of manipulated data that I got from like an example ELISA. And you can see here we use like a monoclonal antibody. So that's an antibody binding to one thing. It prefers to bind a whole sequence and a part of a sequence, and that's the black and the red. And you can see that it also doesn't bind to 2 other sequences. And so from this information, we can see in the 3D structure as well what different patient sera or antibodies bind to. This is a flow chart of my project where I took patient sera and looked at the linear and the conformational or 3D structures of Ig binding to those things, and with collaboration with other scientists and also the other postdoc in the lab, we can use this information to compare between the linear and conformational to get predictive modeling. And I am not the professional on the very complicated coding that he does for this. I know in general, but his name is Dr. Ian Rambo, but he can use this data, and we can help predict whether if your cells bind to this sequence versus this 3D structure, whether you're going to have a peanut or walnut allergy, or maybe you're going to have both. So to conclude, food allergy is a pretty prevalent disease, and it can also be very costly, not only financially or with your life. One of the main food allergies in the US and across the world is peanut, and it's frequently cross-reactive with other foods, and it's nice to know whether you are going to be allergic to another food if you have an allergy to peanut. And so to study this, we look at the different peptides, which are the linear sequences or the 3D structures of the allergens that you are allergic to. And we can use these different methods to help potentially diagnose future allergies. I also wanted to do some takeaways about having a career in science and what to do while you are pursuing your degree. And for me personally, I at the beginning never imagined that I would be a research scientist versus in a doctor's office being like an MD. And so I had to do a little pivot. I had to decide what I wanted to do to help me get the skills so I could become a scientist doing work like on the lab bench and on different molecular techniques. So don't be afraid to bounce around because then you can develop a wide range of skills that you can put on your CV or resume. You never know what's going to pop up in the field because, as we all know, jobs are, well, you want a job and you want to enjoy your time there and keep yourself afloat. And so you want to be able to have a wide range of skills to apply to whatever comes up when you're ready to apply for a job. And like for me, I went from studying plants to using mass spectrometry, which is very analytical chemistry. I did marine microbiology, and now that all culminated into me doing allergy immunology research, and I also teach as well. Another piece of advice I have is to stay connected with other scientists at your institution and in the community in general. Always be ready to talk to even those not in science about your work because it's an important outreach to relay your results to those that might not know all the technical terms. My project here is just one of many in the lab and in the center. And we have a lot of collaborations with Dr. Jacqueline Nesbitt with students at Delgado with the allergy project. I know that she has helped get many volunteers into our lab that have gone on to do careers in like data management for cancer data. That's Gregory down on the bottom left. He was a volunteer in our lab and he went on to find a wonderful career doing data management. We have another volunteer, Angela Graham, and she's working on purifying proteins with Dr. Nesbitt as well. But Dr. Nesbitt's also a really good key contact for the work here. Unfortunately, it's mentioned before that there's a government hiring freeze, and so that even includes volunteers, but they keep saying soon, that will be gone. But when that happens, I would let you guys know immediately. Another thing is always be ready to present your research. So over my career, I've talked at a lot of lab meetings. I've presented posters. I've done oral presentations. I've gone to a lot of conferences, and these things are a little scary at first. And I mean, I still get nervous, even though I talk about my research so much, but it's an important part of growing as a scientist and being able to understand the different areas of research in the world. And last but not least, try to keep an open mind, not only on projects, but different opportunities you find, and also failed experiments, because part of learning in science is getting things wrong and talking to others about what happened and figuring things out. So thank you, everybody, for listening and inviting me here.
Thank you so much for attending and presenting your information. Lauren is a great support and a wealth of knowledge for Delgado and their students. And as she mentioned, a lot of our students do go on to work at USDA or other careers that USDA helped them get into. Lauren, if you can maybe in less than a minute say when you are teaching our general biology lab, which a lot of these students either have taken or will be taking, what are some things that y'all do in the lab that actually translate to what they might see in your lab?
Right. So during like the last, I think, third of the lab that I teach, which is the general biology, we actually do processing of your own DNA and isolating it. And then we're looking at a certain gene, whether you have it or not. And so that includes a lot of molecular techniques, such as, like I said, DNA isolation and purification, and then gel electrophoresis and PCR. So you amplify your DNA and then you run it on a gel, and you can, from that experiment, see whether you have the gene or you express the gene or not. And so that's actually a very hands-on molecular technique that's used in a lot of different labs, like even like crime labs, in plant biology labs. You do a lot of gel electrophoresis and PCR. And so that's actually a really, really key molecular biology technique that you actually get to participate in that lab.
Thank you so much. We appreciate your time, and we
Thank
apologize
you.
that we are running over everyone, and we apologize to you, Lauren, for running over and starting you late.
Oh,
And
it's
this
fine.
allows us to move directly to our next speaker, who is being so kind and patient with us running late. Do a quick introduction. Our next speaker is Dr. Evan Wells. Dr. Evan Wells is an upstream process development scientist at Janssen, and he specializes in cell culture, protein expression, and antibody glycosylation. Sorry, y'all, I'm a turtle researcher. He holds a PhD in chemical engineering from Carnegie Mellon and has extensive experience in the expression, purification, and characterization of proteins in both bacterial and mammalian systems. And Evan's expertise spans bioprocess development, analytical characterization, and advanced protein engineering techniques. Welcome, Dr. Wells.
Thank you for that wonderful introduction. Let me share my screen and get it all set up.
I am going to stop sharing mine if it blocks
Oh,
you.
I think I took it from
That's
you.
fine. Perfect.
All right, and then let me know when you guys can see the full screen slides. Is that everything looks good? Charlene's got the thumbs up.
You're good to go.
Excellent. Hey, well, good morning, good afternoon. I'm not sure if everyone's in the same time zone. I'm in the East Coast, so just welcome to the presentation I put together. I'm very happy to be here today. I was invited by one of your very own Delgado faculty members, Dr. Claire McGraw. We overlapped at Tulane for a while, and we're still best friends to this day. So, happy to have the invite, and thank you guys for bringing me in. And, while I was watching Lauren's talk, I realized I didn't have an about me slide in mind. So I just whipped this together while I was waiting. Just very basic, not as colorful or cool as Lauren's, but, grew up in North Carolina, the eastern part, kind of in the Sandhills region, small town, across the street from a cotton farm. And, didn't really have any kind of, big exposure to like universities or, big tech or anything like that. I just kind of came from a rural area. And I became really interested in school and chemistry and science, and went to East Carolina University, which is, it is a real place. Even though the state is North Carolina, East Carolina University is, while a directional name in the not same direction of the way of the state, still a good school. It's not NC State or UNC, but it's pretty good. And I got my biochemistry and chemistry degrees from there. And I, like many others, probably wanted to go into medical school, but I couldn't get in. And so after I graduated, I worked as a lab tech for a couple of years, just doing protein expression sciences and learning how to purify things. And I got really into it. I started looking into what can I do with a career in this? What are the opportunities? And I realized that chemical engineers do this kind of thing, and maybe I should go become one of those. And so I applied to a group at Tulane University. They accepted me for my chemical engineering PhD. And that's where I ran into your very own Dr. Claire McGraw. We worked together for about 5 years there. And my advisor decided to move from Tulane to Carnegie Mellon. And so I moved up with her to Pittsburgh, Pennsylvania, and finished my PhD in Pennsylvania. And boy, moving from New Orleans to Pittsburgh was a real shock to the system in terms of the weather, in terms of the food. It's a different vibe. But so after that, I moved east towards the Philadelphia area where I live now, and I've been working at Johnson & Johnson, or Janssen, as we've been known, for the last 5 years now. And I've worked my way up from sort of a scientist into a senior scientist, And now my business title is principal scientist slash manager. And I do a lot of work with cell culture. I do a lot of work with proteins, and my main goal is to deliver our portfolio of medicines to patients. And by the way I do that is through manufacturing these and scaling up my cell culture processes to make these therapeutics. And so, what does that exactly look like? If our goal is to use these large molecule therapeutics or these antibodies and other proteins seen here on sort of like the bottom left of the slide, these are pretty big molecules. They're not, they're about 150,000 Daltons big. They're very big. And the way that we make them is with cell culture. So we have cells that produce this. We have to grow the cells in something. That's where the cell culture media comes from. You may have seen this in some of your classes. And to do it in a very controlled way, we have specialized equipment called bioreactors that enable us to scale these things up. Huge amounts of this material can be made at a given time. And the way it kind of works is, you'll have screened, or you might even have, a disease in mind, lung cancer or breast cancer or prostate cancer. And you'll have teams that look for medicines that are useful against those, that target something that's enriched within those cancer populations. So, if cancer cell for lung cancer, produces a lot of, type A protein, we might want to make an antibody against that type A protein that could be effective without killing other cells in the body. So that's the goal here. We start with, one of these desirable targets, generating an antibody for that. Then we make a cell line that can express that antibody. And then we scale it up, we purify it, we make sure it's all nice for patient use. And then we go into regulatory filings and approval. So you may have heard of like the FDA, you may have heard of the CDC, these different like government organizations we have to pass muster with in order to get a therapy approved. And then once we do that, we can go into human studies to test how is it working? Is it effective? And so you might see some of these kinds of proteins on TV when you're watching, the Saints play, or when you're doing something, really anything, watching the Olympics. You'll see anything that's like ending in MAB or MIG. Those are monoclonal antibodies that have been made in this cell culture format. And how does that look? What are the opportunities there? And I think there's a lot of interesting ways that people can enter with a degree from Delgado, or experience and backgrounds that you're getting with your classes there, and sort of work your way into a big pharma role. What I'm showing you here, this is the final product that we make is called drug substance in my group. So that's the stuff that goes into a vial or goes into a syringe that we would then, hand off to a patient for a clinical trial, or, for treating a disease. And, we start with cells in these tiny little vials, and then we thaw them into the media and we just shake them. It's as simple as that. You shake them at a certain temperature in the cell culture media, and the cells start to grow. And as they grow, we need to keep expanding them in order to hit the amounts of cells that we need to make an effective amount of protein. So you can see that we use these essentially giant plastic bags on a heated rocker to expand these cells as up to like 20 liters in this stage. After that, we still need more cells in order to get to the production phase. So that's when we have these giant specialized seed train bioreactors. These range from 50 liters to 1,500 liters for whatever purpose you're trying to suit. But you start scaling it up and getting bigger and bigger and bigger until you get to this production bioreactor stage. And this is a good example of what one of these looks like. It's as tall as you are, possibly taller. Some of these are 2 stories where there are stairs on the side of the bioreactor so you can walk up and look down into it to make sure that it's not foaming too much or anything like that. But after, about 14 days, that's how long it takes the cells to grow in this to express the antibody. We will then harvest it. So if you've ever heard of centrifugation or ever been on one of those carnival rides where you're spinning very fast in a circle, That's what we're doing to the cells. We're spinning them down, and gravity helps pull them down to the bottom, because what we want is in the cell culture media, the liquid that the cells grow in, and not in the cells itself. So we remove the cells at the end of the bioreactor with these very large stainless steel centrifuges. After that, there's an interesting interaction. We rely on nature for a lot of things. It just so happens that there's a protein called protein A, creative, I know, that binds really strongly with a part of the antibody, and is one of the strongest natural interactions found in nature. And we use that bound to basically a sugar bead to purify these antibodies to up to 99% purity in one pass. And it's this, very unassuming, very large looking stainless steel bed that we pump the media through, and it binds to that resin. And after that, we can release it all purified. We have to do some steps to make sure everything is safe for patients by adjusting and tuning to make sure that any viruses that would possibly be growing in there are killed. And then we can store that intermediate until we want to further purify it. And the further purification is mostly to polish up any kind of unwanted impurities, remove any kind of carryover things. Like you had a glass of water, you wouldn't want some dust in it, So that's what we're doing. We're getting rid of a lot of these like impurities that come along. To kind of dive in a little bit more onto some of these individual steps. So like I was saying, we get these cells that thaw into a flask. You can have a big bag full of this like very, unfortunately pea yellow looking cultures. And then this gets transferred into a seed or a production bioreactor. And you can just see how like opaque it becomes. Like these cells are very dense in here. And, there's the old joke that the worse it smells, the more antibody you're making. So sometimes it gets pretty smelly. This is where, so like something like this would be what we'd see at a smaller scale when we're doing just some development work, when you're in the lab, just figuring out the process for the first time. This is the max amount of volume that you would get to. But then when you finally have the process locked in, when you get to the manufacturing facility, you start really scaling up the amount of material, really increasing the volume that you're using to grow cells. And that's when, we keep the flasks in the bags, but these, scale up bioreactors come into play. These production bioreactors come into play. And, one of the interesting things, at least it was interesting to me when I first joined and learned about all this technology, is that, these single-use plastics really help streamline a lot of this. This looks like a big stainless steel container, but inside of it, there's like a soft bag, kind of like the one you see here. That's the big bag that all the cells are growing in to help prevent any kind of like impurities or crossover from an earlier batch. When you use one bag one time and that bag has been validated, you don't have to worry about any kind of thing. You just throw it out at the end, which might not be the best for the environment or the world, but single-use plastics have really sped up how we can do the production of these proteins and medicines in a safe way. So just kind of an interesting thing there. When we get to the production bioreactor, like I was saying, there's a couple of different styles that you can use. We typically have moved more towards what's called a fed batch bioreactor. So like, imagine if you have like, if you're just living your life and you just kind of eat once a day, that's kind of like a fed batch bioreactor. You have like, you only eat breakfast or you only eat dinner, that's just, you give yourself that nutrient feed once per day and you're done, and that would, for these cell cultures, last up to 14 days. That's when we would harvest it, take out the cells and start purifying the media. There are other types of bioreactor runs that, have a more continuous feed. It's like if you're sipping on like, a nutrient shake all day, every time you expended energy, you got new energy coming in. That's what these other types of reactors are. And those can last up to 60 days, but they're more complicated to run. They're more complicated to develop. And so people don't go with those as often anymore because it can be kind of expensive. You have to build a facility around that, which can be billions of dollars. And it's not really the ideal way when you want to have like a more agile portfolio or a more agile set of medicines that you can express and make in a single facility. But I kind of just want to keep commenting on the scale of these things. This is an example of a 15,000 liter bioreactor. It's a lot of stainless steel. You can see all of these plumbing connections require a lot of effort to make sure that they're clean, validated, that they're set up appropriately from batch to batch. And, I think that it is people's jobs to make sure and maintain these and troubleshoot these and, in between clean and autoclave, these kinds of things to ensure the sterility, ensure the safety of these vessels. And, just put it into perspective, 15,000 liters is like a pretty good sized pool. It's 12 by 15 by 4. That's a lot of water. That's a lot of media. That's a lot of cells that we're making at a time. It's a phenomenal amount of power that can go into these. And, at the end of the day, to kind of put that into perspective, if, you have 15,000 liters and you have a pretty good titer process, you might wind up with about 20 pounds of protein, 40 pounds of protein at the end of that could be vialed and shipped out to clinics all over the world for trials or as a commercial supply for patients. So after the cells are done growing, like I said, it's like the teacup ride. We're spinning things, and we're spinning things very fast. About something towards the 3,000 to 5,000 times gravity. So think of one gravity, that's what you are in your chair. 5,000 times that is how fast we spin the cells to get them to stick to the walls of the centrifuge. And this liquid that comes off the top is what we're purifying our antibody from. And after we spin it, not everything gets spun down. Some things are too light to be spun down at the speeds that we work at. So then we have to filter things. So we have, much like, when you make coffee, a coffee filter for your coffee grounds, we have filters that can purify out any kind of like cell gunk or DNA or, other proteins that stick to things that are not good for our process. They get caught to these very large filters that we use. After that, that's when we start purifying over what are called chromatography columns. So just these beads that have an interaction with the protein that we're trying to purify. And we load it on to the bead. And we'll cover this a little bit more in a later slide. But then we wash away all the junk we don't want. And then when all the stuff is washed away, we can then send in a chemical to release that from the beads. And that out comes the purified protein. It's very straightforward. We bind it, we wash it, we pull it off. And after that, we'll make sure, do some viral filtration, make sure that any viruses that are very small and don't get spun down or filtered out. We always try to reduce viruses at any stage we can. We get into other chromatography stages to polish up, like let's say there's something like, you got a clump of protein. You don't want that going in. You want nice single monomeric protein in your therapy. You want to have good product quality. It's a good example is like if you had to make chocolate chip cookies at your house on, Rampart Street, and then I had to make the same recipe of cookies here in Philadelphia. At the end of that batch, I need to have the exact same number of chocolate chips in each and every cookie, the same texture, the same amount of butter. Everything has to match. And that's what we're doing when we make antibodies from a development scale to a manufacturing scale. And then we have to basically file that and pass muster with regulatory agencies of the government to say that this is safe to give to patients. And we have a reason to be licensing this out to use as a therapy. And that's the process. It's very straightforward. We grow the cells, we start purifying with these resins, we filter a bunch of times, and that's how you get a vial of your favorite antibody. Like I was saying, this is kind of what the chromatography stage looks like, where we have a material that binds things. The protein is bound to that material, and then any kind of the junk that we don't want gets washed away. Viral clearance, this is think of like, this isn't what happens, but like, if you have like the flu virus or something growing in your cell cultures, you wouldn't want that getting to a patient. So we have specific types of engineered systems that can remove viruses because of, just some of their structure with like charged parts of their envelopes and things like that. We have a technology that can pull those off to make sure that we're clearing these effectively. And that's what this is. We use several different approaches to that, and those viruses get filtered out. And then at the very end, some of you may have experience or have heard of people who have to get dialysis or have a kidney disease. What we have here is ultrafiltration diafiltration. So this is basically a big dialysis cassette. So like if you ever have dialysis or know someone that gets dialysis, this is what they're doing with their blood. They're sort of rinsing it across this membrane and pulling out and purifying their blood and putting it back into the person. What we're doing is taking it from one composition around our protein into another composition. And that's what this UFDF is. It's just a big dialysis cassette. And this helps us get it to a more concentrated final formulation. And with this chromatography steps, we're utilizing a couple of different approaches. So like I was saying, to bind it, wash things away, then when it's nice and pure, pull it off, and we have a nice pure thing. But then another thing that we'll do is use a resin to polish it up a little bit more, where you can just throw the protein over it, and it doesn't stick to that. It just, anything that we don't want sticks to it. And so that's what the flow through mode does. It allows us to work more continuously to further polish our protein. And I say all this, this is all maybe like high and mighty, like development, and there's a lot of different areas to contribute. So I want to spend some time talking about this. I have a PhD. A lot of my colleagues have PhDs, but on a daily basis, our bench level scientists, our GMP operators, the people who are actually doing the work, they are, we call them our junior scientists. They, come from, a bachelor's degree. Some come from even an associate's degree, where they have different responsibilities and different tasks that they would do within a facility or within a development operation. And so there's a few different tracks that people can take where, manufacturing is a pretty good way to get in. If you're able to find a way to or find facility, these facilities hire people without very much educational background. Like an associate's degree is great, because it's someone that can get in there, be trained up, follow a standard operating procedure, and help make the drugs for the patient, prepare things at every stage of the process, and then run the processes. So there's the manufacturing side of things. Like I was saying, there's the research and development side of things. We have people that come in and are bench level hands that will run our development bioreactors or do our development chromatography steps, and they do an excellent job and are vital members of our team, even if they don't have the high and mighty ivory tower academic background. And then another side of the coin, or another side of the triangle, if you will, is the quality assurance teams. These guys, they really make sure that we're protected from any kind of regulatory scrutiny. They make sure that we do things with high quality. They make sure that things are documented extremely well. They understand what we do scientifically and make sure that when things do go off the rails, which they always do, that we're making sure that we're documenting and rationalizing and using evidence to make sure that corrective actions are placed, that control is maintained, and that any kind of continued issue is noticed and dealt with. And so, yeah, I think that there's different areas for different people with different strengths. And so, definitely look into, potentially, thinking about careers in the pharma industry. There's a lot of things out there, and a lot of things that, whether you're a contractor, whether you're working for a company directly, that, there's ways to make an impact, and, it's not anything that is too inaccessible. So I think I'll stop there and, invite any questions that someone may have. I really appreciate the time today and hope that it's at least a little interesting to some people.
So, Evan, we do have
Mhm.
a few questions. One
Great.
of them is from Christine. I want to say Neeson. If I mispronounced your name, Christine, I apologize. How many times is a product tested before it's released to the public?
Ooh, that's a good question. In our bioreactor manufacturing process, so let me go back to the very beginning. So we have tests that are built in at every single one of these stages. Um, so at the end of the shake flasks, we'll count the cells. We'll see how alive are they. At the end of the production bioreactor, we'll see, okay, the same thing is how much antibody do we make? How is there anything that's growing that isn't our cells in this, like a bacteria? Or, is there any evidence that bacteria was growing in these? So we're testing it there along every single path along the way. And then at the very end, when we get to this stage with the drug substance, it goes through a full panel of product quality, of characterization. And during development, it'll be a much broader set of information that we're trying to collect and gather and organize and analyze before we release this to the public. But then as we develop more understanding and knowledge about the process, you might wind up with a smaller sample set of release specifications that include all of the really important things, but don't have some of that extraneous information that may not be as important. But it goes through a lot of testing on the drug substance side, and everything has to pass within very tight percentages and ranges. So, it can be a little bit challenging sometimes to make sure that when something does look a little bit off, you might lose a whole batch, which could be very time and cost consuming. So, we try to make sure that we build in controls to make sure that everything that we do will meet those release specifications.
Okay, thank you. Another question was about clean in place with the bioreactors and sterilization. Are you trained to do that, or do y'all have a team that's their whole job?
That's someone's whole job. And, it goes beyond just a single team that does that. So there's the people who would actually be in the manufacturing facility doing that. There are people who are on like the analytical side who would then, maybe characterize what are the limits, what are the, potential differences project to project, cell line to cell line that might be a watch out for that team on the floor to adjust how they're doing their clean in place procedures. And, it's goes back to maybe even a third team with, the preparation of the reactors. They work very closely together to make sure parts are autoclaved under, with following the correct SOP or work instruction. And it's just a very tight knit and enmeshed. Many people go into making these drugs. And I think it's a great question. I appreciate that one.
Okay. And then this is just my question. Aren't some of these very similar to what you see in distilleries and beer factories?
Oh yeah, it's you can probably talk to Dr. McGraw about that. When we went to a couple brewery tours back in grad school, she would see me nerding out and asking about like the different centrifuges that they were using. But yeah, to your point, Amanda, they really I think have a lot of crosstalk with each other, and when particularly like I already kind of mentioned the centrifuges, that's a big one for like sort of overlap between brew culture and fermenting antibodies for therapies. But so to that point, yeah, there were a lot of good chats with those guys over the years and a lot of similar materials used.
Do you have time for one more question?
Of course,
Raven?
yeah.
Raven asks, do people in pharma careers often switch between the areas of work within the industry? If so, how easy is it to do? Example, going from the manufacturing to the research side or other way around?
Oh yeah, it happens all the time. I think, very frequently, I'd say. One of my, managers, he started as a drug substance manufacturing operator on a drug called Remicade back in like 1996. And, he didn't have a college degree at the time. I think he just had a high school education and, just kind of, worked at it, went to night school, eventually got his bachelor's. And, now then like after a little bit of time, he switched from manufacturing over to the development side. And now he's a director level person in our group. There are other times when people might, kind of the manufacturing hours can be long and arduous, you know, but over time, that might not be fit for your work-life balance that you're trying to achieve. So you might go towards more of a desk type job or a development type role where you have a little bit more opportunity to control your own schedule. And so, yeah, it happens all the time. I can think of 5 or 6 people in the last couple of years, even that have had similar transitions between quality or manufacturing and development. And it's, I think, a way to expand your breadth and really just bring different perspectives into different areas where, someone that is in development may not have that quality perspective, or someone in manufacturing may not have that development perspective. And so switching can be really beneficial for a company too, depending on what you're interested in. So don't hesitate. I think that trying things out is always really cool.
Well, thank you
Yeah.
so much for your time. I know we're way over what you were expecting, but
Well,
we appreciate
hey, I appreciate it. Yeah. Well, I hope that, you guys enjoyed, and, if there's any questions that come, please feel free to reach out, and, look forward to chatting.
Thank you so much, Evan. We appreciate you. And I'm going to go to our next speaker.
Awesome. Thanks, guys.
Thank you. Our next speaker is Dr. Thomas Tubon. Dr. Tubon is a national leader in biotechnology education and workforce development, and he currently is serving as Executive Director of the Technology Career Institute, that's a long name, at Mira Costa College, where he leads industry-driven training and community education initiatives. He previously served as Chief Workforce Development Officer for Biomade, guiding national programs to strengthen their bioindustrial manufacturing workforce. Before his leadership roles, he spent over a decade as a biotechnology professor at Madison College, where he led multiple NSF ATE initiatives advancing STEM education. And he holds a PhD in molecular genetics from Stony Brook University and Cold Spring Harbor Laboratory. I will say that Dr. Tubon is a longtime supporter of Delgado and has done many stem cell educational research classes for our students and faculty over the years. So no further ado, go for it, Tom.
Thanks, Dr. R. And it's great to see everyone here. And thank you for the invitation to speak with you all today. I've been with Delgado Community College SLT program since 2016 as a strong supporter of everything that you do. From its initial inception to the growth, I've seen every step along the way, and I'm nothing less than impressed. And so I'm going to outline a little bit of a different talk here for you all, and I want it to be a little bit more interactive. And so I'm going to ask the students who are here and the potential students for the SLT to weigh in from time to time. And so, this is a very interesting spin on career development pathways. And it's relative to the science lab technician, the biotech programs, to all things related to STEM careers. And so I'm hoping to share a little bit of insights into this whole idea of, where are you now and where are you going to go to? What lies ahead for you all? And where's biotech going? And so this is kind of scoping out this entire thing for career pathways. And Dr. R kind of outlined a little bit of my background in career pathways, and it's convoluted. So it's a little bit more like the pathway on the right. But, as students go into the academic environment, many are faced with the choices of what the heck am I going to do, right? I mean, I can go into medicine, I can go into nursing, I can go into engineering, I can do all these different kinds of things. And oftentimes those things will change. So don't be surprised if your pathway forward leads to a different route, because as long as you're doing what you want to do, you're always going to be doing the right thing. The pathways that we take for our careers are always filled with stops and goes. And this is a reminder to say that there's no wrong pathway to take as long as, again, you're doing exactly what it is that you want to do. Now, I'll tell you, in all transparency, my career began in the sciences when I was in 7th grade. And I took a science for, the College for Kids? You all have College for Kids out there? Okay, so this is like, you put a 7th grader into a college class and you put them into anatomy and physiology, and they get to, look at cadavers and do all really kind of cool things. And it really kind of sparked an interest in what's possible in science and such. And so by exploring a lot of these different things, I decided to move that pathway forward. Now, where I started is not where I ended. And as Dr. R mentioned, I currently oversee programs that are in welding, that are in engineering technology, and in drone operation for the FAA, our heavy equipment operators for building highways and stuff. And it's like, okay, well, what does that have to do with biotech? It has everything to do with biotech because it's analytical thinking, it's reasoning, it's what you do to solve problems. It's engineering and thinking your way outside of the box. And that's one of the core things that this education in the SLT program really kind of helps people to develop, that can do mindset. And so just keep that in mind as you're going through your pathway. This is kind of a snapshot of my life journey in technology, and I'm not going to go into all of these different things, but one of the highlights that I will mention, and this kind of came out as a staggering out, like a realization for myself, is that I've been in this field for 36 years. And so I've seen this come from very few, 2 or 3 biotech programs in community colleges, now to over 100 different programs that are out there through our community colleges across the US. And I've had the fortune of working with many, many, many of them and seeing their programs develop and flourish, like the one that we have here at Delgado Community College. So I'm going to ask you all from the audience side, a survey was recently done, and this is talking about careers, right? STEM education and careers. And they surveyed all the presidents and the executive folks in these higher education institutions, and they said, how many of you guys really think that the people that you're putting through these programs are prepared to go into the workforce? And the response from the higher executives, and I'm one of them now, was 96% believe that they were pretty strongly ready, right? What do you think from your side the actual answer is, from the employer side? And if you can enter that into the chat box, I would super appreciate that. Dr. R and Ms. Shenik, I can't see any responses. Are we getting anything in?
Come on, students, don't be shy.
You could do it. You could do it. If someone says 96%, I want to go to your school. 1 or 2 responses? Any responses? How many of you think that from the industry side, the hiring people in industry, they see people coming in and they say, oh, yeah, this percentage of people are ready to go into the workforce? Okay, I'll give you guys a spoiler here. When they look at the industry and the business leaders, 11% of the business leaders say that those who have trained in academia in these programs and STEM programs are ready to go into the workforce. So there's a lot of work that needs to be done out there. But fortunately, Delgado SLT program has tapped into the main pipeline for how to solve these problems and integrate those things that the industry is looking for. Your partnerships with industry, your partnerships with all of the employers are really strong, and they're the driving force behind why the program is so incredibly successful. Why is this so important? Well, the estimation is that there are 25 million positions in science and technology enterprise, which are going to be coming out in the next few years. That means that there are more jobs that are going to be out there than there are going to be people to fill them with. And so this comes to you as students going into science and laboratory technician type programs, right? We heard a lot about the therapeutic side, and those were amazing talks that we just heard from. But we want to make sure that as people are going through these programs, everyone's prepared and they can hit the ground running when they enter a company like Johnson & Johnson or, enter into an R&D environment in education, that they know their stuff. And I've come to know the instructors and the Shenik of the programs, and I can guarantee that the experience is top notch and it is aligned with our industries. So there's no falter there. Okay, so I'm going to take a step back. I am not in New Orleans. I'm in Southern California. I'm in San Diego. I'm on Oceanside, about a mile away from the beach. We have a huge biotech program here as well, one that offers a bachelor's degree in the community college. And we're going to see more evolution of these programs that are aligned to the industry. Now, we're not an isolated type program here, and our sister program is up in Solano Community College, up in Northern California. But the network of all the community colleges work tightly together. And I'm going to tell you guys a little bit about that, because when you come in as a student to some of these programs, we have spent the last 20 years building the network so that these resources are not just an entity like Delgado building amazing things. These are shared resources that speak to the value across the United States. So these are the 4 things that the National Science Board recommended. And I'm pointing these out because as part of my role in the last 20 years, I've gone everything and played every role, from speaking at the White House for manufacturing to working at the front lines in biotech programs and developing programs for stem cells and regen med. But at the end of the day, these 4 suggestions were put out there for why and what we need to do to be more effective programs. We need to change the message. You're in a community college, you're taking a technical program, and I cannot tell you how many times in my career I have heard people say, okay, community college, keep an eye on that. You should be targeting a 4-year institution and a bachelor's degree, a master's degree, PhD. The general need right now, and the biggest need in changing the message, is that we need skilled technical workers out there to do the work on the production floor. When our previous speaker spoke about the SIP, clean in place for those fermenters, we need people who are dedicated to do that, who know the ins and outs, who know how to fit those pipes and know the impact for when things go wrong, troubleshoot. So we need to change the message. The value of these programs needs to be elevated, because without that, a lot of these jobs and programs and everything are going to be shuttled overseas to lost opportunity for the United States. We need to focus on the data, and the data says that programs like Delgado's SLT program should be at the front of the line. We need to leverage the federal portfolio to help support programs like this. And so Amanda and Charlene, this means we need to find ways that we can help you guys secure money so that you can elevate and innovate for the program, get some new equipment and build those partnerships. And so that's the last one, build the partnerships, because everything is based on the partnerships in the industry that you all bring in. So all of these 4 things are roles that I play as part of what I do here at my college, but also on a national scale. And so this is one of the reasons why I remain very closely tied to Delgado and other community colleges. The network that's out there. There are 136 community colleges that are part of something called Innovate Bio. And some of you may have heard about this, and it's part of this. Delgado is a strong member for Innovate Bio. And this allows resources that are coming in, not just from Delgado, but from everywhere. So you can see that a lot of folks are putting wind in the sails for success of the SLT program. We have worked together on a number of National Science Foundation sponsored things like workshops and such in stem cells. We've worked with cross-institutional student networking, which was a lot of fun. Bringing students from my institutions in connection with Delgado students for meetings and conferences and for cross collaboration. And then other areas that lead to program expansion, like memberships in Biomade or some manufacturing advanced technology with Georgia Tech, NSF-ATE, things that you hear about that contain the program content, but allow resources to expand beyond the borders of any particular region. What that means is that you have access to a lot of different resources as part of that SLT program. And your leaders behind this are the ones who are opening those doors for opportunity. A couple of pictures I wanted to share with you about our students and some events that we've had in the past. And you can see the middle row all the way to the right is our illustrious Ms. Chenik doing amazing things in the laboratory. And so, these kinds of things, I'm not going to get so much into the equipment operation, the biotech, the molecular biology, all of that stuff that you cover in the classroom, right? I want to focus on the outputs and stuff on the user experience here. But we've had a lot of these interactions, and the program at Delgado is incredibly strong, like I said. So let's take away all of the molecular biology, all of the instrumentation, all of the different things that we know the program teaches to, and ask, what are the core skills? What is industry looking for, right? Scientific literacy, how do you prepare something? How do you prepare a buffer or a reagent, a solution? You're going to be using something. How do you document what you do? What are your techniques to do that? How do you calibrate different things or use different pieces of equipment? right? This is a short list of what the program teaches to, and it's been one that's identified through other partners throughout the United States. Now, this has been memorialized, and it's not just, spewing out what's required for science technician core skills. We actually published a paper not that long ago, and we quantified all of these different needs and demands. All right, this is a complicated slide. I don't expect anyone to read this one through. But these are the things that people teach in biotech: documentation, cell biology, molecular biology, cool stuff. There's nothing on this list that you all aren't going to touch in the
The
time that you're in.
slide's blank. All we're seeing is a black
No.
screen.
Okay,
Yes.
how about now?
No.
How about now?
I can see it.
Okay, then maybe it's just
Charlie, can you see this slide? It looks like a Christmas tree or whatever. It's red and green. Can you see this one?
Okay, it's my computer. Sorry, everybody.
Okay. Okay, so these are biomanufacturing knowledge, skills, and abilities, the KSAs. Okay, this is great. Why am I pointing that out? Because when you're done with your academic training and your readiness for work, this is what the industries are going to be looking for. When you apply to them, they're going to say, hey, do you match these criteria? And if your answer is yes, that's going to elevate your candidacy for getting a really amazing job in the biotech field. All right, I don't want you to look at the different things. It's complicated, like I said, but what I do want to distill this down to is when we look at this, and when I look at this, and I published this paper a few years ago with Wake Forest, with the folks over at Wake Forest Institute for Regenerative Medicine. And the conclusion is that these skills, which are all the way on the right, the red bars that are important but not available, all pinpoint to our need to address biomanufacturing and the creation of product. Now, that's important because that's what all these techniques and stuff kind of lead up to, or many of the techniques will lead up to, whether you're working with DNA, whether you're working with animal models, yeast, or whatever. Ultimately, for biotech, it's about what product are you going to make. Stack on top of that product interpersonal skills. You're going to get an earful of this in the future, so I'm not going to spend too much time on this, but this is also an important part of how people really kind of work together in the industry side of things. What's important? And so I'll share these slides with you, and you can take a look at these a little bit closer. Again, this is the national network. You're now part of it, and so just thinking about what kind of resources, what kind of friends do you have across the United States? And you can find and have Dr. R and Charlie help identify some of these partners that are across the US. All right, I'm going to take a switch really quick because I got 2 more minutes, right? I got 2 minutes. I'm going to take these 2 minutes. I'm going to pivot a little bit because all the things in biotech where you're talking about pharmaceutical products, where you're talking about, how do you create different things, are really important across the board. And I want to ask you guys another question here. You guys can enter your answer into the chat box. How many of you are entering into biotech because you want to go into medicine or some kind of health or some kind of pharma? You want to improve the human condition by health? All right, I'm going to have to lean on you all for any type of responses I get to that. I'm going to pause a second.
Use your
My gut feeling
reactions
tells
if
me
you want to. Or
100%.
no, yeah, 100% because no one's responding.
Okay.
Go ahead and use a thumbs up if that was your goal. Okay, we have Raven said that would be her focus.
Okay, cool.
We have Samantha wants to be in a subset, but Regina and Christine do want to be in the medical realm. So 3 of the 4 want medical and 1 of them wants a subset.
All right. Well, I'll say almost exclusively when I ask this question to folks and to students, almost 100% say, yeah, I want to go into pharmaceuticals. I want to go into medicine. I want to go into therapeutics, which is absolutely fine. And I think it's a great thing to go into. Part of that is new technologies and such, but there's also another arm of biotech that people are not necessarily aware of. And this is more on the manufacturing side for things like, you ever think about the buildings that we live in and producing the cement for those buildings using bacteria, right? Is that even possible? All right. So the Department of Defense has developed technology so that you can actually spray bacteria onto like sand, and those bacteria will cross-link and form cross-links to solidify that sand and create what is stronger in its durability than concrete, right? So you can create housing materials. You can create all different kinds of things. You can use this type of technology to create things that impact food and ag and biofuels and fabrics and textiles, right? I like my shoes. I like my leather shoes, but I know that there's a push out there to create synthetic leather using mushrooms. That's biotech, right? Aquaculture, our fisheries. In San Diego, 88% of our seafood is imported. What can we do to use the power of biotech so that we can create aqua fisheries? Right? And so those are interesting new questions that impact the future of biotech and these future jobs that are coming out. Now, is it an impactful thing in Louisiana? Absolutely. So up towards Baton Rouge, there's a company called Origins that's building out a power plant or manufacturing plant that has those fermenters at scales of 100,000 to 200,000 gallons, as a matter of fact, to produce things like commodity chemicals, to produce things that we use every day that's not necessarily in the medical area. Anyway, I know I'm rambling on here. This is something that I'm incredibly passionate about, but I wanted to kind of open up the door to a couple of different conversations and pathways moving forward. And I know I'm at time here. And so I want to respect everyone's time because we are over time. And I'll open it up to any questions that you all may have. And thank you for the opportunity to be here with you all.
So I see that we have a student that said they're keeping their options open. If any of you have questions about what are some possible alignments, Dr. Tubon has so much experience and exposure working with so many different fields that he could probably show you a link between what your degree is going to be in and how to get into that field. So think outside the box and use these networking opportunities while they're in front of you. Do you use AI? That's from Christine.
AI is a relatively new entrance into the game for biotech. And it usually is something which is going to it's going to be something which impacts the production floor. AI increases the efficiency. AI is not going to get any dumber. And so if we don't embrace AI in the production and the use of biotech, it's going to be a missed opportunity. Let me tell you one use of AI that's really cool. There are 4 factors that they have identified that reprogram cells to make stem cells, right? They used AI to actually computer design more efficient molecules and manufacture and how to manufacture. And they actually did it, and it works a lot better. So, you know, AI, it'll come, and it's going to be an important part of biotech.
Great question, Christine. And Venetia, you are on a great track. I personally know your track. So Venetia stated, I love where I am right now in my Chemtech journey, but do want to branch off someday into something new.
Cool.
Any other questions for Dr. Tubon? Well, thank you so much. We appreciate your time. Again, sorry that we did run over, but we got just a wealth of knowledge from every individual that stayed and presented. So we appreciate everything. What I will say to everybody is once this recording encodes and we get a transcript with it, which could take hours because it is 3 hours long, this will be sent out to everyone that shared their email address with me. Then you can share that you attended with your instructor if needed. Any slides that were shared with me from the presenters, I will also send in that same email. If you have any questions, you can contact Ms. Shunik, myself. I will also send the slide emails with the slide email about the SLT program, so you have that as a reference also. So again, we appreciate your time and your commitment, and hopefully you learned something that will help you move forward in your career path. Any last words, Ms. Shunik? Okay. Well, thank y'all very much. And just look for an email from Dr. Rosenzweig, A. Rosen, coming up in the next day. Thank you, everybody. We appreciate you attending.
Thank you.
Awesome,
Sorry,
Samantha.
I thought we had one more speaker, but I realized he's presented with Dr. Roberts.
Yeah, we
Robertson.
combined them. They requested for combination. I'm going to go ahead and end it, Charlie, and start encoding. Bye, everybody.
Thank you.